And let me go ahead and answer some of these questions for you guys. Again, this is obviously the game plan we are talking about here. Today is Thursday, April 23rd. We're going to continue our discussion of the nervous system. We talked and laid the groundwork for it. So today we're going to talk about producing graded and action potentials. And then we should have some time at the end to start talking about brain anatomy. Again, you have the list of all the assignments. There are 4 more assignments due in this section, 2 more unit reviews. Your PhysioX exercise 3, which again is not hard, but has 9 different activities to it. So it does take a lot of time to do that properly. And then you do have that 15-point nervous system review, which will be graded for correctness. It will be available today, but like I said, I want you to encourage you to not start working on it yet. We need to at least talk about the anatomy of the spinal cord before it will really start to make sense to you. Because the point of it is to emphasize that relationship between structure, function, and location. And so one of the keys to this, if you know one, then you should know the other 2. And so you'll actually see that on the review. On the review, as you're doing the review, you'll see, well, this is kind of a redundant question. And yes, that's the point. Because like I said, if you know the structure, you should know the function and its location. If you know the location, you should know the structure and the function. So if I give you one of those 3 things, you should be able to infer the others. And so that's what it's about. So there is some redundancy on those questions, and that is on purpose because it is trying to show that relationship. And again, that will be graded for correctness. All right, now the things that started this, got a couple questions. One of the things that I find we are having less time for as we go through this, because the lectures are taking a little bit longer in this format, is for the histology. And one of the things that I asked of the classes is to give me questions or comments about the format of the exam. And some of you did that obviously during the exam, but others as well. And one of the things that repeatedly has been pointed out to me is that the histology feels a little bit lacking. We haven't had as much time for it. And so what I am going to do, especially because there's a fair amount of histology on this one, and it could be a good component, like I said, maybe 20% of our lab exam. We're not really having the time to go over it in class, so I want to have an opportunity to be able to go over it together because I think that is some of the more challenging stuff. To help with that, I am going to have a histology review this Friday from 11 to 12. I know that is outside of our normal class time, so I will be recording it and I will post it onto, post that onto YouTube, but I strongly encourage you to be there so that you can ask questions and make sure that these things make sense, because going back and viewing it is not going to be as beneficial. And as a carrot, because we don't have open lab and things like that anymore, I will provide some type of extra credit provided if you do come to that, to be able to to participate in that. So if you come for that, I will provide some type of extra credit. But whatever that extra credit is, whether it's 2 points or 5 points or whatever it is, you will gain much more by understanding the histology, and you'll get more points by being successful on the exam than you will from those extra points of extra credit. So come for that, look at it for that. So that will be available on YouTube. I will post it sometime after I do it, but I strongly encourage you to be there. Speaking of exams, let's talk about the last exams. The last exams were hard, apparently, and there was a bit of struggling on it. They have been curved. The curve is a bit challenging on Canvas. Canvas was not set up for this. Proctoria was not set up for this. The way their exams are set, the point totals are locked. So I can't curve them the same way I would curve them normally. So instead, what I'm doing, again, to take the lab exam, normally whatever the top score on the lab exam becomes the top score. That becomes the 100%, and we go from there and get a percentage. So what I have done instead, if you'll notice in the grade book, is that rather than curving the grades, I have figured out what the curve would essentially be point-wise to get a similar percentage based on if we had curved it. And I have added the points in as basically what shows as extra credit for that. So again, your percentage that you show for your grade is really those 2 scores together divided by whatever the total is. And if we do that for the lab exam, we see it's a little over 60%. If we see it for the lecture exam, it's also a little over 60%. Those are not great totals, and we need those to come up significantly. The good news is that with the additional work we've been doing in classroom and lab-related activities, the overall grades are still decent, 77, a little over 77% for those. But again, I fully expect that to come up. All right, the other thing that I want to talk about the exams is I want to talk about the exam policies. Again, one of the things that I have tried to emphasize a lot in this class, and I guess I'll have to sneak this in here, is that obviously we need to maintain the integrity of the class. That is why we are doing things like the Proctorio and all of those types of things. You guys now have that under your belt. Again, one of the things that I've always said that I like about this class is that this class is hard. I don't have to do things to make the class more challenging. I don't have to be tricky on the exams because this class is hard. And I don't want the exams to get in the way of you showing me you have the information. And I appreciate for some of you, the exam format did get in the way of you showing me that you had this information. But what you have to realize is, unfortunately, because of these unique and unexpected circumstances we're in, this is going to be the format for the rest of the class. So hopefully having that experience will help you to be more successful and get past that and be able to show me your knowledge. The other thing, though, is exams should be a learning experience, right? And I want you to be able to learn from it. So after class today, I purposely did not do this before class, because if I did it before class, you would all be looking at it and you wouldn't be any paying attention to me at all. But after class today, I will be releasing the exams for review. This will give you an opportunity to be able to see the stuff that you got wrong, figure out where you lost points, and be able to learn from the experience that way. These exams will be available for only 24 hours. Actually, slightly less than that. Basically, at 11 a.m. tomorrow, when I start the histology, no, actually, today's Thursday, yeah. So when I start the histology, I will be relocking the exams. All right, so that gives you an opportunity to be able to look at the material, learn from the material, help yourself to be successful moving forward, see what it was about the process that challenged you, and see if you can find ways to get by that and really focus on presenting that information. So I want that to be an important learning process. And so I think this is the best way to do that. There's been a tremendous amount of discussion. I know this is new for all of you, and there are some challenges for that, but you have to realize this is new for me as well. And so I think I talked about this in the last class. Other instructors and I, the entire biology department, the entire A&P departments, have had many meetings, many discussions about what the best way to handle these types of situations are. And based on those discussions and my own personal preferences, I think this is the best way to get the best of both worlds, to help to maintain the integrity of the class and the exams, but also to allow these exams to be a learning experience for you. So I think that is going to be important. And so for now, that will be the policy, unless I find people abusing that policy, in which case it'll be taken away from you. So that is important as well. And that actually brings me to my future exams. Because as I talked about, we talked about this, I believe, in the last class as well, but it is something I definitely want to make sure that we emphasize and talk about here. On the future exams, the remaining lecture exam will be the exact same format as the one we just had. No new information, no new processes. There will be multiple choice, there will be fill in the blank, there will be essay questions. They will all be presented at once, and you can scroll through it and complete it. You will have a set time limit just like before. Everything on that will be the same. There's nothing new on that as well. For the lab exams, as I think I told you last class, I'm going to be guinea pigging my 431 class because, as we talked about in the last one, most of the problems that people had, and there were not a lot of problems, there were only a few, but the few problems that we did have, most of them involved the lab format. So the lab format, I am going to, for theirs, because their next test is actually Wednesday next week. For their lab exam, I am going to allow them to see it all at once and see how that works, if there are loading problems or other issues with that. And based on those results, it will either be the same as the last test, or it will be the same as the lecture exam. Right. So again, the good news is there's no new process there. I'm not changing the process. I'm just going to use one of the two processes we have already established. So it will be one or the other. So again, nothing new on that, and I will let you know ahead of time which of those two formats it is going to be. The last thing is the final exam. We don't want to be thinking about the final exam yet, but we do have a final exam. I have no idea what the format for that final exam is. I have yet to decide whether it will be all multiple choice questions, whether there will be essay questions. If there are essay questions, will there be a study guide available? Will I provide that study guide? Will you provide the study guide? There's a lot that goes into that decision. There are a lot of logistical issues with that. So don't ask, I don't know. Again, last week, you had your exams. My other class had their exams. I literally finished grading your exams yesterday. You probably saw when I started posting the curve. And then I've already started preparing for our histology review to get that stuff settled. And then my 431 has their exam next week. So this weekend, I plan on doing more research. We have another department meeting on Friday as well. So I hope to have a better idea of what the format for that final exam is going to be. So I have no idea right now of the format for it. I do know it is going to be cumulative. That is still going to be the same. And the other thing that is going to be the same is there will still be a benefit for doing well on the final. It counts as a grade on its own. And again, that's important. It still counts as its own grade. So even if you're getting an A in the class, you still need to take the final. The final still counts as a grade on its own, but there will also still be a benefit. Now, because of our change of format, I do not know if it will be a straight replace, or if, depending on how well you do, there'll be certain bonus points that you can get added to your score from that. But there will be some kind of benefit for doing well. This class in particular, again, I didn't pick the things that we're learning randomly out of a hat. We are learning this material because you need to know it for 431. And so retaining this information, gaining this information, retaining this information is going to be vital for your success in 431. And so to strongly encourage you to not just cram the information in and purge it on the screen and then forget about it, but to retain this information to help you be successful in 431. I want to reward you for being successful at doing that. So there will be some benefit for doing well in the final exam. I'm not certain what that is. Obviously, it'll depend on the format of the exam as well. So we will see how that works out, and I'll have more information for you on that next week. All righty. Any questions on any of that? All right. I know there is at least one or two questions that were being asked about last week's, last day's content. So if you don't do well on the final, no benefit. Well, so, okay. Think of it this way. And again, all I can speak of is to what it would be in the normal class. In the normal class, remember, you have 5 lecture exams, and then you have your final exam. And let's go really simple, right? I tell you, you can replace one of your lecture exams with your final exam. If on all 5 of your exams, you got a 90% on all 5 of them, and on the final, you got an 88%, do you want me to replace one of those 90s with an 88? No, of course not, right? So again, if your final exam score is lower than your lowest lecture score, then replacing them doesn't help you, right? But if you struggled on that first exam because you didn't know what anatomy and physiology was going to be like, or maybe you struggled on that third exam because it was when we were transitioning online, or maybe you struggled on this muscle exam because it was the first section we did all online, or something like that. But you gained the information, you retained the information, and you showed me you can be successful with it on the final, then I allow you to replace that exam with the final to benefit you. So again, because I know that life happens, I know learning happens, I know there are bumps along the road. So if at the end you reach the goal of gaining that information and being successful in the final, then I give you a reward for that. I don't throw out exams. I hate people who do that. That's like saying one of the 5 sections of your class isn't important. You can just ignore muscles because you know that's going to be the exam you throw out and it's not going to affect your grade. Now, that's not how this works. You have to earn that reward by showing me you've gained this information. And if you've done so well that you don't need the reward, then bonus, right? Or if you totally bomb the final, you don't want me to replace one of your scores either. But if you can do well in the final and you struggled somewhere along the way, then you get an added benefit for that. That's how that works. And something similar will happen in this class as well. All right. All right. Any other questions on the things that I've talked about so far? All right. I know Esther at least had one question on last lecture's content. So if you want to go ahead and ask that, we can do that. And then if anybody has any other questions, I can answer that. And then we'll move on to the new material.
So my question was in reference to the beginning of the lecture where you're talking about structures of the brain. You said that the central nervous system and the peripheral nervous system, those are functions?
No, those are the two structural divisions.
Okay.
So all of the structures of the brain are divided up into 2 parts, just like all the functions of the brain are divided into 3. All the functions of the brain are either sensory, motor, or integrative, right?
Okay.
All of the structures of the brain are divided into 2 parts. Either they're divided into the central nervous system or they're a part of the peripheral nervous system.
Okay. And so the brain, the spinal cord, those are structures of the central nervous system, and nerves, ganglia, plexus, and sensory structures are structures of the peripheral nervous system?
That is correct.
Okay. Even though the central nervous system and peripheral nervous system themselves, those are structures as well?
Those are the structural divisions. Think of it this way. The skull, the spinal cord, or the vertebral column, the bony thorax, those are all structures that are all part of the structural division of the skeleton, that is the axial skeleton. Right?
Okay.
That makes sense?
Yes.
It's the same thing here. You have, or heck, let's be even simpler. You have a table that is made of a tabletop and 4 legs. You have a central nervous system that is made of a brain and a spinal cord.
Okay. So you said for the central nervous system, it's command center? It's the integrative?
Yeah, it is an integrative function. The command center, remember, was a term we used when we talked about homeostasis. But when we really think of its function is integrative. But
okay,
if you think back to homeostasis, that's really what we talked about. It receives information, it processes that information, makes a decision based on that information, and then sends the information out. Well, that's integrating. That's integration of information. So really, it's not new information. We've just added a new label to it.
Okay. And then you said peripheral nervous system is sensory.
No, peripheral is sensory and motor.
And
there
motor.
Are 2 branches of peripheral. There is the afferent, sensory coming in, and the efferent, motor going out.
Okay. And then you said we're talking about the little mini braids of plexus.
Mm-hmm.
Those are structures though too.
Well, be careful. They're not mini brains. What they are is elaborate networks of nerves that can do some basic processing.
Okay. I will just cross it out so I don't get confused.
Yeah.
Okay.
But yeah, a plexus is just a fancy word for an elaborate network.
Okay. If I know that you're doing the Zoom classroom tomorrow, if I have more questions, can I ask you some of those then?
At the end, if we have time left at the end of that, I want to focus solely on the histology. I don't know if it'll take an hour, but if there's more time at the end, or if you have other questions, then we can meet outside of that time in my office to discuss these things as well. I'm always available by appointment in my office for Zoom meetings.
Okay.
Okay.
All right. Thank you.
Yep. One other question that came through by chat while that was going on, just as a reminder, for the histology review, in your module, under your lab handouts, you have a histology list, the list of all the histology that you're going to be responsible for on the exam, and that's what we're going to go over. We're going to go slide by slide, item by item through that list. So I would encourage you to print it out so you have something to look at. I mean, I guess you could bring it up on your screen or maybe even on your phone because, you know, Canvas has an app. You could bring it up on your phone and be looking at it there while you're looking at your computer. But I think if you have it printed out, then you can actually be doing some of the drawings while we're doing it, or at least make notes on it and do the drawings after the fact. You don't have to complete the histology before. If you want to complete the histology before, you're welcome to, because that could help to see if you got it right. But it's you're not turning it in for a grade. Although maybe that's how I'll provide the extra credit. Maybe I will have you guys fill it out and turn that in. Maybe that's how I'll do the extra credit for that. Maybe I like that. That's good. I got to make a note of that. Yeah, because that way I don't have to worry. Because then that way people who aren't able to make it during that time don't get punished for that as well. So just like we do the reviews, you fill them out, take a picture, and submit them. I'll do that, I think, as the extra credit. So that way you're not required to do it, but if you want to do the drawings and turn them in, that is smart. I like that. Thank you. Good idea. That helps everybody. So yes, yeah, I would encourage you to do that. That helps you to study. You have it to study from, and that way, if something happens during that time, or you're not able to make it because of work or family or anything else, those people aren't punished by that. And so that will be. I'll just tell you right now, so that you have the weekend to work on it. We'll make this the handout will be due. So the histology. And I will be due, let's go Tuesday. And so Tuesday is with the 28th. Yeah. Tuesday the 28th at the beginning of class. Perfect. So that way, if you're not able to make it to the meeting, you can at least watch it. I've added a new website to our list of tools as I was hunting through Google, basically just Google images to find histology. I kept again and again being sent to this Yale histology site, which is freaking amazing. So I encourage you to take a look at that. I've added that link, and at least a third of the pictures I have so far have come from that. So that's going to be something that'll be really useful as well. So, so that it can help you with your drawing, as well as you know, you have your professional anatomy lab that we have. You have your textbook, your lab manuals, all of those other things, and even the CRC has some histology there as well. Under the modules at the very top, the first unit is study tools or useful tools or something like that. I added it there, so you should be able to see it there. There is a fair amount of histology on this lab exam. Like I said, probably, I haven't done the math, but my guess is easily 20%. All the spinal cord stuff we're going to talk about, the cerebellum, all of the structure, function, and location of nerves, a lot of that can be done histologically. So yeah, there will be a fair amount of histology on this one. My guess is probably somewhere close to 20%. So the list you have is not extensive, but there has some, it's a good amount. Again, 20 to 25% cranial nerves, depending on how much sensory stuff we get to. And we're already behind on lecture, so I don't know how much sensory we'll get to. So no, the extra credit won't be for attending the histology review, but for turning in the handout. Because again, I don't want to penalize people who aren't able to make that time. That is outside of our normal class time. I don't want to require people to have to come outside of their normal class time. So is the D test going to be longer? I assume that's lab. I assume we're asking about the lab test. No, I say no. I would say that the lab test is smaller. I mean, the muscular system is our biggest. There's the most anatomy on that. This one typically has 60 to 70 questions. So it's more similar to what we did on the first two tests. All righty. Questions on any of that? All right, I think we have covered everything then. So let's go ahead and dive into the lecture. I will stop distracting you with my magnificent beard, and we will get on to the lecture. So let me go ahead and put that there. Put that there. Go ahead and turn that off. And we can switch gears and get started. So bang. All right. We left off last time, and what we were talking about is that how we get cells to do work. And the way we get cells to do work, as we talked about, is basically by changing its membrane potential. The cell, it wants to be happy, and it wants to stay, oh, where's my annotation tool? There it is, at its resting membrane potential. And what did we say that resting membrane potential was again? One of the important numbers you absolutely have to have for this class, absolutely. Negative 70 millivolts, excellent. Again, remember at that point, it's not that there are no ions moving. There is plenty of ion movement into and out of the cell, both passive and active. Leak channels are opening and things are sneaking in and out. Pumps are being used with ATP to move things in and out. But the key to this point, as we talked about, is there is no net movement. The overall change, there's no, or again, another way to think of it is there is no change, right? As we talked about, if one positive thing comes in and one positive thing leaves, or one negative thing comes in and one positive thing comes in, right? One positive cancels out one negative, one positive leaving cancels out one positive coming in. There is no change, and that's equilibrium, right? That's the key. That is our equilibrium. Oh, that shouldn't be there. I don't know if I can cut and paste that. Let's find out. Yes, I can. Cool. There you go. That's our equilibrium point. But again, that's our cells equilibrium. And that's what our cell killer is about. However, ions are 2 years old, 2-year-olds, and all they care about is themselves. While the cell wants to be at equilibrium, so does sodium, and so does potassium, and so does chloride, and so does calcium, and so all those things. And so that is their driving force. Their driving force is to move either into or out of the cell to reach their equilibrium potential, to make the cell equal to their equilibrium potential. And so, of course, if we want to know how an ion will move, we need to know the equilibrium potentials of those ions. And while every ion has one, the two we are going to be responsible for, the two most important ones for us, are sodium and potassium. And as we talked about, the equilibrium potential for sodium is positive 66 millivolts. So what that means, when we open a gated channel, when the sodium channel opens, sodium enters the cell, right? And that makes it more positive as it is trying to reach its equilibrium potential. Conversely, whoops, no, wrong button. Potassium is negative 90. So when we open a potassium channel, potassium leaves the cell. And when it leaves the cell, that makes the cell more negative because it is trying to get to its equilibrium potential. And even though we don't need to know our equilibrium potentials, we certainly know which way does chloride move when it goes, does it go into the cell or out of the cell? When you open a chloride channel, which way does chloride move? Right, chloride goes in. So that must mean its equilibrium potential is lower. Which way does calcium go again? Right, it goes out. Excellent. Right. So again, we don't know their equilibrium potentials. Well, we know them. I know them. You don't need to know them. But again, the equilibrium potentials is what gives these ions their driving force, determines which way they want to go because they want to be at equilibrium. And as we talked about in the last class, that's important because when an ion, which is a positively charged ion, oh, great question. Let me finish this first. Actually, I'll answer your question first. That's a great question. So the cell wants to reach its equilibrium potential. But if you think about it, if we have a big, huge, massive cell, and we open just 3 sodium channels, do you think enough sodium is going to be able to come inside to be able to drive the cell all the way to its equilibrium potential? No, probably not, right? If we opened 1,000,000 and we kept them over for a long time, then yeah, it would probably eventually reach there. But as I'll tell you, the sad story of it is in normal physiology of our neurons, these ions never get to their equilibrium potential. They try their darndest to get there, but none of them are going to actually get there. And that's good for us. That allows us to do work. Because the cell wouldn't be happy at positive 66 millivolts. So we don't want them to be successful. So yeah, so they will try, but they're not going to be successful. They will try though. Great question. I love that. Excellent. So, like I said, these ions, whether it's sodium, potassium, chloride, calcium, all of these things are charged particles. And when a charged particle crosses the membrane, either going in or going out, it's moving. And that movement is a current. All right. It's like the same thing as that electron we talked about at the end of last class, going on that wire to your light to light up that light. That is a current. And those currents, as we talked about, are our potentials. And as we talked about in the last class, there's not one, but 2 types of potentials we need to focus on. So let's go ahead and continue on from here. That was all really review. We'll start first simple with our graded potentials. Remember, as we talked about, graded potentials are going to be small deviations from the resting membrane potential. Again, some of the information we talked about them is that, again, they're small in amplitude. They, on average, are around a 2 millivolt change. They can be slightly bigger. They can be slightly smaller than that. And that's one of the other keys. They are variable in amplitude, where they can be a little bigger or a little smaller. And the other big key with these graded potentials is that they can be positive or negative. Now, we know we are going to get a graded potential by opening channels. And when we open channels, we let ions flow. Right? We can actually draw this, and normally I would do this on the board, but I think we can do this here. I need this up out of my way. There, excellent. So down here we have time. Over here we have our voltage. And as I mentioned, there are some important voltages we need to know. One of those important voltages is up here. Another of them is, let's say, right here. And again, these may not necessarily be drawn to scale, but it's okay. We'll get the point. And let's put the other one down there. Those voltages, as we know, are the equilibrium potential of potassium, which we know equals negative 90 millivolts. Up here, we have our equilibrium potential of sodium, which we know equals positive 66 millivolts. And then the other important one we need to know for right now is our resting membrane potential. And of course, we know that equals negative 70 millivolts. Now, as we talked about, if we leave the cell alone, it will happily stay at that negative 70 millivolts forever. But we want the cell to be able to do work. And so the way we can do that, and let's start with blue first, is by, let's do it all the way, opening channels. All right, to do work, we have to open channels. One example of this would be if we opened, and I think I have to change my font to get a little teenier so this all fits in. Let's say we open 10 sodium channels. I assume you guys can read that okay? Maybe some of you on your phone may not be able to read that okay. It's not too small, is it? Yes, it's too small? No, not too small. Okay, perfect. Excellent. If we open 10 sodium channels, sodium is going to be able to rush in, right? Its driving force is to want to enter the cell. And sodium enter the cell makes the cell more positive. All right, we're comfortable with that. That's something we've talked about before. Right? And that's exactly what we would measure on this type of a graph. What would happen is our resting membrane potential would go up as sodium was entering the cell, making the cell more positive, and trying to get to its equilibrium potential. And what did we call that kind of type of process when a cell gets more positive? We say the cell does what? There you go, excellent. The cell depolarizes. Excellent. Now, with only opening 10 sodium channels, it isn't necessarily going to be enough to get it to its equilibrium potential, but we will get it more positive. Right? But these types of channels typically don't stay open very long. Maybe it was a chemically gated channel that we opened, right? And the acetylcholine diffuses away, or the enzyme breaks it down, or something like that occurs. So eventually what happens after a brief period of time is the cell goes back to rest. Our cell goes back to rest. Of course, the question is, how does it do that? Oops, I need to make that smaller again. The question, how it does that using pumps to move things out, and also our leak channels are going to play a role in allowing the cell to go back to, and let's be more specific, to resting membrane potential. And if you remember back to the last section, we had a name for that when you go back to the resting membrane potential. And what was that name we gave to when you go back to your resting membrane potential? Repolarizes, absolutely, or repolarization. I'll be consistent. The cell repolarizes. But again, depolarization, repolarization, that would have been fine as well. All right, think about it, really no new information. That's kind of something that we already understood. But what can also happen instead is instead we could open 10 potassium channels. And if we open 10 potassium channels, what's going to happen then? If we open a potassium channel, which way is, you absolutely have hit it with the right word, absolutely. In this case, but again, the immediate effect of this is that potassium leaves the cell. And when it leaves the cell, that makes the cell more negative than the resting membrane potential. And when the cell gets more negative than the resting membrane potential, you guys have hit it on the nose, that process would be what we would call a hyperpolarization. or hyperpolarizes. Again, let's be consistent with the terminology that we have been using for all of this. Hyperpolarizes, excellent. Sneak that above there. I think that works. Perfect. Once again, it is not going to stay hyperpolarized forever. Those channels are going to close. Once again, the cell goes back to its resting membrane potential. And again, it does this with pumps and leak channels. And what do we call this process of the cell getting more positive, going back to our resting membrane potential? Oh, you're right. I did, no. It typically, if I open just 10 potassium channels, it's not going to reach equilibrium potential. So I apologize for that. I just overdrew my, so here, let's cheat. We'll bring, no, that you get to stay there. You get to come down here a little further. You get to come down here a little further. You get to come down here. You get to come down here. And I need to add to my line. Thank you for catching that. I apologize. No, I purposely don't want to reach the equilibrium potential. So I apologize for that. Yeah, opening just 10 is not going to be nearly enough to reach the equilibrium potential. So I apologize for that. There, I cheated and fixed it. Excellent. Thank you for catching that and asking that question. So let's go back to my question. What do we call this process of going back to the resting membrane potential when we've been hyperpolarized? Well, it turns out we call it repolarization. To repolarize means to go back to rest. It doesn't matter what direction. Notice it doesn't matter the direction, right? A depolarization, and let's use green for this. A depolarization is when we move more positive than the resting membrane potential. A hyperpolarization is when we move more negative than the resting membrane potential. But repolarization means to go to the resting membrane potential. So it doesn't matter the direction it's going, as long as you're going towards the resting membrane potential, we consider it a repolarization. All right, you guys with me so far? We had to add, excellent, we need to add two more things to this. The first, remember, these graded potentials typically occur, let's do it this way. I'm going to change the color again. Let's not even say typically. Occur on the cell bodies and the dendrites. And remember, it is our cell bodies and dendrites that are not specialized the same way that the axon is. And remember, the other key to this is that this is variable. Right? That is one of the key things to remember. So if you think about how we could possibly make this variable, well, notice the first time we opened 10 sodium channels. But what if instead we opened 20? If we opened 20, then the signal would be bigger. If we opened 100, then the signal would be even bigger. If we opened 3, then the signal would be even smaller. Notice as we change the number of channels that open, we change the amount of sodium that comes in, and we get a bigger or a smaller signal. And it works the same way with the potassium channels. We could open 5 potassium channels or 3 potassium channels, right? So that's 3, that's 5. We could open 20. Again, I want to avoid the equilibrium potential. As we open more and more, or less and less, we can change the size. And so then the last piece we need to add to this then is what type of channels must we be using? If you remember, there are 3 types of channels. Gated channels. What types of gated channels, right? So when we talk about the gated channels, remember there are 3 types. There was chemically gated. Oops. There is voltage gated. And there is a mechanically gated. So for these gated channels, what are the types that we could use for something like this where we could only open a few? Well, let's talk about chemically gated channels. With chemically gated channels, is it possible to maybe just release 2 acetylcholine and get 1 sodium channel? Or maybe if I release 100 acetylcholine, 15 sodium channels, and if I put 1,000 acetylcholine, 47 sodium channels? Notice with a chemically gated channel, we can change how much chemical is present, and that will change how many number of channels open. So absolutely, that works. We could use chemically gated channels to be able to get a variable signal. But what about voltage gated? Voltage gated have a magical point where they open. What was that magical point where our voltage gated channels open again? We call it threshold. Absolutely. You guys both hit it on the head. We call it threshold is that magical point, and you are absolutely correct. Threshold, and I'm just going to write it T here, equals negative 60 millivolts. The thing with a voltage gated channel is that when it reaches threshold, it opens. So if we get the cell, and let's go ahead and draw our line here. Draw in pink. If we were to get our cell up to negative 61 millivolts, how many of our voltage gated channels would open? Half of them? A third of them? Well, it's not that they're not controllable, but they're controlled in a different way, right? Let's think of it this way. If I have, let's say I have 100 voltage gated sodium channels. If I get the cell up to negative 61 millivolts, how many of those 100 voltage gated sodium channels are going to open? 10's a good guess. Nope. The answer is none of them. None of them open. Why? Because I haven't passed threshold. If I pass threshold and get to negative 59 millivolts, how many of those 100 voltage gated channels are going to open? All of them. That's correct, right? Voltage gated channels are all or nothing. You can't get half of them to open. They have a threshold. So if you're below threshold, they're all closed. If you're above threshold, they're all opened. So there's no flexibility in that. Again, it's tremendous control. We got plenty of control, precise control, but they're all or nothing. So notice we can't use voltage gated channels for a we can't use a voltage gated channels for oops, I did that. Let's go back. Hold on. Made a mess of this. All right. Notice we can't use our voltage gated channels to get a variable signal because they're all or nothing. And if you think about it, all or nothing sounds an awful lot like what our action potential is. And remember we said our axon is specialized? Well, guess what our axon has that makes it specialized, that makes it produce a big positive all or nothing action potential? It has voltage gated channels. Now, let's think about chemically gated channels. Mechanically gated channels. Mechanically gated channels, you stretch it and a channel opens. So if you stretch it a little bit more, do you think we'd open more channels? If you stretch it a little bit less, do you think you'd open fewer channels? Yeah, with mechanically gated channels, the more you stretch it, the bigger your signal you get. The less you stretch it, the smaller of a signal you get. So notice when it comes to our graded potentials, when it comes to producing graded potentials on our cell bodies and our dendrites, our cell bodies and dendrites are going to have chemically gated and mechanically gated channels on them that allow us to get both positive or negative depending on what type of channel they open, and bigger or smaller depending on how many channels. And that's really the key to a graded potential. There are two rules for graded potentials. All right? The direction is determined by the type of channel. Right? If we open a sodium channel, the cell gets more positive. If we open, so sodium and calcium, if we open sodium and calcium channels, we are going to get a depolarization. Whereas if we open potassium or chloride channels, we get a hyperpolarization. So the type of channel we open determines the direction. And the other rule is the size, or if we want to be fancy, the amplitude, is determined by the number of channels that open. The more channels we open, the bigger the signal we get. The fewer channels we open, the smaller the signal we get. No, mechanically gated and chemically gated are two different keys. Right? Something that is mechanically gated is a channel that opens or closes by the stretch of the plasma membrane. So you change the shape of the plasma membrane and you can get that to move. It's like I said, the example of squeezing your arm. If you grab your wrist and squeeze your wrist, you can feel that pressure change on your wrist because of those Pacinian corpuscles, those laminated corpuscles that are in your skin that you're changing the shape of. When you squeeze the skin, you're changing the shape of the skin, they're being stretched out, and you get a signal sent to your brain telling you, hey, someone is squeezing my arm right now. And hopefully you're aware of the fact that it's you, right? However, chemically gated is like when you take the sip of coffee that's sitting behind you right now so that you can stay awake during the lecture, right? You get the bitterness of the coffee or the sweetness of the sugar, right? Those are chemicals binding to the taste receptors of your tongue, sending a signal to your brain that you're perceiving something sweet. So they're both channels that can be opened and closed, but the keys to unlock them are different. Just like a voltage gated channel is a channel that can be open and closed, but it has a different gate and a different key that unlocks it. And that one's a change in membrane potential. All right, great questions. Any others on this? Okay, great question. So what I would say right now is that we aren't getting to the point of producing an action potential yet. Right? But that is the last piece. Actually, that's actually really the last piece that we have to do with all of these things. Notice the goal, and let's write this up here at the top, and we'll pick a color we haven't used yet. What color haven't used yet? Doesn't look like I've used yellow. Does yellow show up okay? Our goal, oops, so I spelled goal correctly, of our graded potentials is to produce a signal that spreads to the axon hillock of the neuron, and there we determine if we fire an action potential or not, right? And that is really the key to this. These are our decision making. One of the things, remember, we talked about is that I took up more space than I thought I was going to. So I need to move this a little bit. And I'll take a picture of this so that we can save this. Right. One of the things we've talked about at the beginning is our brains are not hardwired together. Our neurons make decisions, right? Your house is hardwired. Every time you flip that light switch on the wall, your ceiling fan turns on. You flip it the other way, the ceiling fan turns off, right? On, off, again and again and again and again. There's no decision making going on. That is an advantage to that. The advantage to that is definitely speed, but that wouldn't really help us from a nervous system standpoint, because you want to be able to make decisions on how you respond, right? I'm sitting here in my computer, in my desk, in my house, far away from all of you. So if I pick up, right, my Rubik's Cube or some other toy that I have by my desk, and I, in a very firm voice, say, I'm going to kill you, all right? That's not going to change anybody's behavior while they're sitting there listening to this. But if someone busts open your door, and the person who busts open your door happens to be a bear holding an axe, and that bear holding an axe looks you in the eye and says, I'm going to kill you, you are receiving the exact same stimulus, but you want to respond to it very, very differently. We don't want our nervous system hardwired together. So what happens is our cell bodies, our dendrites, receive signals, and they send those signals to the axon hillock, and depending on the number of signals and the size of the signals and the direction of the signal determines whether or not you fire an action potential, whether or not you respond. So if you think about it, if we send a big signal, a 30 or 40 millivolt positive signal towards the axon hillock, that's more likely to fire an action potential. If, on the other hand, we send a negative signal, well, that's going to take it farther away from its threshold, and it's going to make it less likely. So positive and negative signals here in our graded potentials, basically, really what our neuron does is it counts beans. How many positive beans are there? How many negative beans are there? And based on that, it makes a decision. And so this is how we're able to modify our behavior, is by producing different types of graded potentials. All right, Oksana, did I answer your question? I see, Gabrielle, I see yours as well. Give me a second to make sure we answered this one and we move forward. We'll talk more about the action potential next. And again, we'll emphasize that more when we get to that action potential next. But the short version is, you're absolutely right. A hyperpolarization is going to make it harder to produce an action potential. But there's sometimes we don't want to, right? We'll talk about this more when we get to the spinal cord. But if you think about reflexes, if you are sitting on that, again, assuming you could go to doctor's offices now just for a normal checkup, if you were sitting on that bench in your doctor's office or that table, exam table, one of the things he'll do if it's just a normal checkup is hit you in the knee with a hammer. And when he hits you in the knee with a hammer, your leg kicks out, right? Now, again, and it's a reflex, you do it without even having to think about it. However, if they then put a sleeping baby in front of your foot and hit you in the knee with a hammer, how many people are going to kick the baby, right? Only probably about 3 of you, because some of you are just mean. But most people will stop it. So being able to stop those action potentials and stop that behavior is something that is important. All right. Can you please explain why the voltage gated can't be used? Yes. Voltage gated channels are like the light switch on your wall. Again, most light switches have 2 settings, on and off. There's no in between. So if I have that light switch, and again, if you think one of the old-fashioned switch type light switches, right, you put a little bit of pressure on it and nothing happens, and a little bit more and nothing happens. But if you put enough pressure on that switch, it flips from off to on. It has 2 settings, off and on. Okay? Now, if you hit it twice as hard, the light doesn't come on twice as bright. Or if you only put it halfway up and hold it there, the light doesn't come on at half intensity. It has 2 settings, on and off. You flip the switch on and it's on. You flip the switch off and it's off. And that's what a threshold is. The threshold is the point where the gate opens. If you are below threshold, the gate is closed. If you are above threshold, the gate is open. It only has 2 settings. And every single one of our voltage gated channels has the same threshold. Notice if I have 10 chemically gated cation channels. If I have 10 channels that all open to acetylcholine, but I only release one acetylcholine, it can't open all 10 channels. It can only open one. Then if I release 3 acetylcholine, it opens 3 channels. If I release 9, it opens 9 channels. Notice how much acetylcholine I release determines how many of those channels that's open. I can open 1 or 2 or 5 or all of them. It doesn't work that way with voltage gated. I can't just get half of the voltage gated channels to open. If I am below threshold, all of them are closed. If I'm above threshold, all of them are open. I can't change them. I can't I can't vary them. I can't change how many open. It's either all or nothing. And remember, that's the key with a graded potential. It is variable. The signal can be smaller or it can be larger. And I can't vary how many voltage gated channels are open. Gabrielle, does that make sense? Awesome. Great questions, you guys. I really appreciate that. It really helps me to just, sometimes when I'm sitting here, it seems like I'm talking to a brick wall. It gets rough. So I really, really appreciate it. I know this is more challenging stuff, so I know you guys have questions. So I really, really appreciate when you guys do ask them. Again, if you don't want to type in front of everyone, just send it to me privately. But I love it when you guys ask questions. I really appreciate that. So thank you. That means that you're listening. And because I know this stuff is confusing. And I promise you, if you're confused, somebody else is as well. So please ask the question. All righty. Excellent. So those are our graded potentials. Question on that? Any other questions? All right. So here's what I'm going to do. I'm going to minimize all my windows because since I'm not on the whiteboard, when I take a picture, it shows all of my, like the chat window and the participants and all of that. I'm going to take a picture of this because I think this is worthwhile. So let's save this so that we can put it on the screen. Then we'll go through all the pretty pictures on the words for it, and then we'll switch gears to our action potential. So let me put all of these down and out of the way, and then I can put this. I think there works. Oh, I actually have to bring back up my annotations. Perfect. All righty. All right, so that's saved the screen. So I've saved that. So now I can go ahead and clear it. And now we can go through basically all of it again right here. So again, graded potentials only occur in the dendrites and the cell bodies, and they're going to vary in amplitude. And the reason for this is it uses either chemically gated or mechanically gated channels. And now, so that is the reason for that. And now I think we can switch back to black to make that a little easier to read. So they're going to vary in amplitude because we use chemically gated and mechanically gated channels. And here, as we see, that depolarization is moving away from the resting membrane potential. Repolarization is moving back to it. A hyperpolarization is moving below the resting membrane potential. And again, that return to the resting membrane potential, and I don't know why they didn't write that there, but remember, if you're returning to the resting membrane potential, this also is a repolarization. Oops, it helps if I don't do that. All right, so there you go. Now, these are tiny little waves you're producing in your pool, right? So again, we're all stuck at home. So hopefully you have a nice pool out in your backyard. So you can go out of the backyard. You can sit down at the edge of the pool, or really lay at the edge of the pool, put your hand on the water and just lightly slap the water. And as you lightly slap the water, you will produce little waves. You can slap it a little harder. You can slap it a little lighter so you can change the size of the waves. But if you're like me and you have that massive Olympic-sized pool in your backyard, okay, I don't have any pool in my backyard, but let's pretend I had an Olympic-sized pool in my backyard. If I'm just there slapping the water at one side, are those waves going to make it all the way to the other side of the the other side of the pool? No, of course not. They're not going to make it that far. They're only going to be able to travel try that again. They're only going to be able to travel short distances. But the good news is that's all we need. We only need them to reach the axon hillock. Because once they reach the axon hillock, they can determine if we fire an action potential. So luckily, they don't travel far, but we don't need them to. We just need them to get to that axon hillock. And if they can get to that axon hillock, then we can determine if we fire an action potential or not. Because it's going to be these graded potentials that determine whether we do. All righty, that is our graded potentials. So now our goal is to switch gears and talk about the action potential. I'll leave this picture here for right now. I know we're not as far in the lecture, but again, we spent a good 20 minutes with the introduction today, which I'm totally okay with. Like I said, I want to be fully transparent in this process. I don't want this process getting in the way of you guys being successful in this class. So I know this is a challenge, so I have no problem spending that extra time at the beginning to make sure that we are comfortable and ready. But this is a good natural breaking point so we can come back and handle the big, mighty action potential. And so we will talk about that. Let's go ahead and take our first break. It'll be a 10-minute break. I have no idea where my phone is. So I will pretend that I know what time it is. Looks like it's about 1:07. And so let's come back. There you go, 1:06. Let's come back, meet back here at 1:17. And at that point, I will start the recording. All right, I'm gonna go ahead and pause the recording now. Everybody go get some coffee, get something to drink, get something to eat, take a shot of alcohol, whatever it is that you need to get through the next hour. But let's go ahead and do that. All right. So let's talk about a action potential. As we kind of were hinting at, action potentials are large deviations from the resting membrane potential, and they only occur on the axon. Remember, we said the axon has a specialized plasma membrane that allows it to produce and spread, or again, the fancy word, propagate an action potential. There is no new information there, but in the last part, we kind of hinted at what the new information is. And the reason it is able to produce and propagate that action potential is that the plasma membrane, or again, remember the special term is axolemma, has a massive number of voltage gated channels. And in particular, two types. Those two types are, and again, since I already wrote voltage gate, I'm going to abbreviate it VG, sodium channel, and a voltage gated potassium channels. Now, both of these are voltage gated channels. Because they're both voltage gated channels, they have the same threshold. So again, for abbreviation, T is going to equal negative 60 millivolts. And I'll do the same thing here. T equals negative 60 millivolts. That part is the same. But there are two big important differences between our voltage gated sodium and our voltage gated potassium channels. Voltage gated sodium channels open and close quickly, whereas voltage gated potassium channels open and close slowly. The other big difference, and I'm running out of room, so maybe if I do an ampersand here. Voltage-gated sodium channels have that ball and chain. Yes, so the threshold for both is negative 60 millivolts. Did I not say that? Yeah. So both are going to open at negative 60 millivolts. If you're below negative 60 millivolts, the channels are closed. If you're above negative 60 millivolts, the channels are open. So yes, both have the same threshold. But the speed at which they open is different. Voltage-gated sodium channels open right away. And when they pass negative 60 going up, and when you pass negative 60 down, they close right away as well. Voltage-gated potassium channels, when you pass negative 60, take a lot longer to open. And when you pass negative 60 going down, it take a lot longer to close. The other difference between them is voltage-gated sodium channels can inactivate, whereas our voltage-gated potassium channels don't. Remember, we talked about this in the last class. Some channels have extra proteins. And remember, we called it the inactivation gate. But remember, that inactivation gate was basically two proteins, a globular protein and a linear protein. So they're also called the ball and chain. And those can wedge themselves, those can block the channel when it is open. And if it blocks the channel when it is open, ions can't flow through, and they are inactivated. Voltage-gated potassium channels do no inactivation. So they don't have that same ball and chain on the potassium channel. So those are the big, that is what's special about the axon, that it has those voltage-gated channels, massive, massive numbers of these voltage-gated channels. And there are two types of them. Our voltage-gated sodium, which open and close quickly, and our voltage-gated potassium, which open and close slowly. Sodium have three states, open, close, and inactive. Potassium have two, just open and closed, because sodium has that ball and chain inactivation gate, and the potassium ones do not. Because we're using voltage-gated channels, remember, it's all or nothing. Right? This is an all or nothing system, or nothing signal, really. Either all the channels open or none of them open. You can't get half of an action potential or 3/4 of an action potential or 1.5 action potentials. An action potential is a set amplitude thing, one or nothing. And remember, we said it is about a positive 100 millivolt change. So it is a huge, big, positive set signal. That big positive depolarization of 100 millivolts. And the advantage of that is it is able to then travel very long distances. Instead of you standing at the edge of your Olympic-sized pool and slapping it with your hand, instead you get one of those Fiats, right? Because those are something that you could lift up over your head. You get one of those little smart cars and you throw it into the edge of the pool. You are going to get a big, huge, massive wave. And that huge, massive wave is going to be able to travel long distances. However, going the 3 feet necessary to get from the small of your back down to the big toe is still a really long distance. And again, if you think about that, you take that small car, you throw it into the edge of your pool, you're going to get a massive wave, and that massive wave is going to spread a long distance. But it is going to get smaller while it travels. And that's one of the special things about our action potential. Our action potential actually self-propagates, or really the other way we can think of it is it self-strengthens. What happens is the action potential as it travels, unlike that wave in a pool, which gets smaller as it travels further away from you, the action potential stays the same size. So the whole journey of that action potential stays the same size. And we'll actually talk about how that occurs a little bit later. All right? Excellent. All right, so as we have often done, oh, so I do actually have room to leave that. No, stop it. I want to leave this here. As we did in the classroom, when we have some of these more elaborate processes that we did for the muscular system too, we pulled up the whiteboard and we went through it together. For this, I want to do the same thing. For this first one, again, and especially because you have the chance to go back and watch this the video, I really want you to just sit back, watch, listen, think about what we're talking about, all the stages, all the immediate effects. I'm going to do the whole entire thing for you on the screen, and then we'll come back and do the whole thing again, all pretty, all perfect, with all of the pretty pictures and all the pretty words afterwards. So we'll go through it twice. All right, so let's go ahead and do that. I'm going to switch gears and switch from this to our whiteboard. And then here on the whiteboard, clear what we had from before. We certainly need our graph. So let's go ahead and do that here. And again, down here, we have our time. Here we have our voltage. And as we know, there are, as we've talked about, 4 important values we need to know. And I fully appreciate that these are not going to be drawn to scale with each other, but that's okay. It's the point. Oh, my voltage is off the screen. That's interesting. Barely there. Top of this is not really the top of this. There you go. Excellent. All righty. And these 4 values that we most know are, of course, the equilibrium potential of sodium, which we know is positive 66 millivolts. We know our equilibrium potential of potassium, which equals negative 90 millivolts. We have our resting membrane potential, and again, for space, I'm just going to abbreviate that, resting membrane potential, and we know that's negative 70 millivolts. And we know there is a threshold for our voltage gated channels, and we know that is negative 60 millivolts. And remember, this is the threshold for all of our voltage gated channels, both, and let's even emphasize that in writing. This is the threshold, and I think I'll make it a little smaller to make sure it fits in here, for, oops, both the voltage gated sodium and the voltage gated potassium channels. So all channels open, all of our voltage gated channels open at this threshold. All right, that's going to be our graph that we are going to need, but there's one other thing we need to draw to help us to remember. And let's go ahead and sneak in the space over here, our neuron. There, of course, is our neuron cell body. We know that it has the dendrites that are on it. And we know it has one nice big long, actually, let's do it this way, axon that will come to axon terminals where it branches and does all of that stuff. But the other thing we need, and that for this we'll need to change colors, so let's go here. As we talked about, we have this transition area of our axon known as the axon hillock. Oops, it's supposed to be a return. There we go. Remember, this is the trigger, this is the transformation area where we get from the specialized plasma membrane of the axon to the non-specialized of the cell body. Remember, and again, I'll just draw a bunch of them here. Our voltage-gated sodium channels, our axon has massive numbers of voltage-gated sodium channels, again, along the whole entire length of this, all over. It's filthy with them all over the place. And again, this is where we start to get the formation of those, that transition area. And as we talked about, we know there is some place inside of this. We don't know exactly where it is, but we know there is a point inside of our axon hillock where we can produce our first action potential. And of course, we call that the trigger zone. Excellent. So what we are doing, what we're actually measuring here is this would be like if we had a voltometer where we basically put this voltometer right here in the axon hillock and we were measuring voltage changes. This is basically what we're doing. We're measuring the voltage changes that are happening in this cell just at this one location, just in the axon hillock, just at this trigger zone. And of course, we know at rest, and we can continue to use red for this, this is going to stay happily at negative 70 millivolts and will stay there forever unless we mess with it. All right? So this is our starting point. Okay? The last thing we need to remind ourselves of is, as we mentioned, here, starting at the trigger zone and moving down, we have massive numbers of our voltage gated. So let's actually write this down here. We have our voltage gated sodium and channels and our voltage gated potassium channels. And when the cell is at rest, both are closed. There. That is our starting point. All right. Excellent. So the first thing that needs to happen, and let's go ahead and write this here, is that we receive and I'm going to abbreviate it, graded potential from the cell body. And I'm going to write cell body, but remember it could have come from the dendrite as well. So what happens first, and again, let's use our pen, and we'll use pink to represent this, right? Somehow we released, for instance, acetylcholine in this area. We produced that graded potential. And that graded potential, as we talked about, spreads down the cell body, spreads down the dendrite, and reaches the axon hillock. And we perceive that as a small depolarization. Now, if that depolarization only gets us to negative 68 millivolts, does anything else happen? No. As we talked about, that's going to go back to its resting membrane potential and will continue there for forever. That's not very interesting. But maybe we get another graded potential. And maybe this one's a bigger signal. Or maybe we even have a third one down here that's even closer. The point is, what happens is our graded potentials come down here. And if our graded potential, and I'll switch back to red now, if the graded potentials, oh, I want to write it out again, are big enough to reach threshold, that is when the magic is going to happen. Right? If we're big enough to reach threshold, this is when our magic's going to happen. This is when we get our action potential. And specifically, if it's big enough to reach threshold, the important thing to remember is that we'll trigger the opening of all of our voltage-gated channels. All right, so here is our threshold. We've reached threshold. That is big enough to reach threshold. All of our voltage-gated channels are going to open. However, remember, not all of our voltage-gated channels are the same. Both of them start the process of opening at negative 60 millivolts, but do they both open at the same speed? It's one of the easy questions. Give it to me. Do both voltage-gated sodium and voltage-gated potassium channels open at the same speed? No. Which one opens first? Not calcium. There are no calcium. We've got voltage-gated sodium and voltage-gated potassium. Sodium, absolutely. So what happens first is those fast voltage-gated sodium channels open. And remember, not just this, but all open. And remember, we have massive amounts. So we get the massive opening of a massive amount of voltage-gated sodium channels. Massive amount of them open really, really quickly. And so the immediate effect of that is we get a massive influx of sodium. A massive amount of sodium enters into the cell. Sodium, of course, is a positive ion. So we get a big positive depolarization. Or let's say it this way, because positive means depolarization. Big, rapid depolarization. Right? And guess what? A big, rapid depolarization, this is the action potential. We get our action potential. Accomplished our goal. End of story. Okay, not really. But we get this rapid depolarization as the sodium is entering the cell. Because remember, sodium is trying to reach its equilibrium potential. Sodium is trying to reach its equilibrium potential with that massive influx of sodium. All right, we've got our big positive signal. We've got our action potential. But as we've kind of hinted at, when we're going to use a muscle, do we just want to send it one action potential? No. It's not like this one neuron produces one action potential and then it gets to retire and go to the old folks home. We need to be able to produce many action potentials. So not only do we need a big positive signal, but we need to recover from it so we can do it again. And so while this here in our literally second step, we have gotten our action potential, the process still continues. Now, as I mentioned, sodium is trying to reach its equilibrium potential. Remember, that's positive 66 millivolts, and I know it's sitting right next to it, but I'm going to go ahead and write that anyway. But it does not reach it. Instead, it peaks around positive 30 millivolts. So we add a fifth number to our list. That fifth number that we are responsible for, that fifth voltage that we are responsible for, is going to be the peak of the action potential. Oops. And that peak of the action potential, the fifth, and I'll tell you right now, final number you need to know is right there, and it equals, oops, need to fucking negative, pardon me, positive 30 millivolts. Now, the question becomes why? Why doesn't it reach its equilibrium potential? Why does it peak at 30? And the answer for this is because basically does not reach the equilibrium potential of sodium for two main reasons. The first of these is those voltage-gated sodium channels. Remember, those voltage-gated sodium channels opened really quickly, but remember, as we talked about, they're not stable in that open state. So what happens is the ball and chain, or really the ball of the ball and chain, wedges itself into the voltage-gated sodium channel. And again, what that means is the channel becomes inactive. Remember, when the channel is inactive, even though the gate is open, sodium cannot flow through it. What this means is no new sodium can enter the cell. And if no new sodium can enter the cell, it can't depolarize anymore. All right. Now, that would mean that it might just stay up here depolarized for a long period of time, but that's not the case either, because a second thing occurs at about the same time. Remember, way back down here, we pass threshold, and threshold are when all of our voltage-gated channels open. So the second thing that happens is those slow voltage-gated potassium channels finally open. Took them a while to get there, but they finally opened. And let's think of the state of the cell at this point. Potassium, there's a lot of it inside the cell, very little outside, so it wants out of the cell. Potassium is a positively charged ion, and the cell is positive. And positive things don't like being where it's positive. So if you think about it, at this point, sodium really wants out. And so now that these potassium really wants out. Now that we finally open those voltage-gated potassium channels, and remember, there is a massive number of them, we get a massive efflux. should be 2 F's there, I think. Potassium. All right. Lots of potassium leaves the cell. And of course, if lots of potassium is leaving the cell gets more negative. Right? Because a positive ion leaving the cell makes the cell more negative. Right? And so the key to this is not only does it stop getting more positive, but now the cell, oops, that's not a now. This cell is going back towards resting membrane potential. So the cell repolarizes. And so we've reached our peak of 30 millivolts, and now the cell starts to repolarize. Now, in fairness to potassium, at first, the cell is indeed repolarizing. But is that really what potassium cares about? No, potassium cares about its equilibrium potential. So is potassium going to want to stop at the resting membrane potential when it gets there? Nope, it's going to want to keep on going. It's going to want to keep on going and trying to reach its equilibrium potential. Because again, our driving force is the same. Potassium wants to reach its equilibrium potential. So not only does it repolarize the cell, but it actually takes it beyond. It hyperpolarizes. So not only does it repolarize the cell, but it hyperpolarizes the cell as well. This portion where it goes beneath the resting membrane potential, we call this the undershoot. Right, it undershoots the resting membrane potential and just keeps on going. Because again, what potassium cares about is reaching its equilibrium potential. So it doesn't care about the resting membrane potential. We should actually draw the resting membrane potential. Let's do it as a really fine line. So there's our resting membrane potential. So we have a point of reference. Let's also put our threshold back in there as a point of reference as well. All righty. And I'm going to cheat and move this down to where we actually pretend like we registered there. Grab that. Oh, not you. I want you. I'm cheating. I don't care. There. Now it's prettier. Perfect. We've undershot. Now, I'll tell you, give you a hint. We don't reach threshold. We don't reach the equilibrium potential of potassium either. So we do not reach the equilibrium potential of potassium. But we haven't necessarily explained why. And the reason why actually takes place right here. Notice at this point right here, something special is happening. At this point right here, as the cell is repolarizing, because again, it is still a repolarization, it's still going back towards the resting membrane potential, it passes the threshold on the way down. As the cell repolarizes, it passes threshold on the way down. And as we know, that closes all of the voltage-gated channels. Now, of course, as we talked about, the voltage-gated sodium channels close quickly. And something else important happens. Remember, when our voltage-gated sodium channel closes quickly, that kicks the ball out, and the cell is no longer inactive. Or let me say that, the channels, the voltage-gated sodium channels are no longer inactive. All right. However, as we talked about, let's actually emphasize that by doing that. Notice our voltage-gated potassium channels close more slowly. And because they close more slowly, this is why we get the undershoot. Because as long as they stay open, the cell is going to try to reach its equilibrium potential. Right? Notice we don't reach the equilibrium potential because eventually with a delay, the voltage-gated potassium channels close. And of course, once they close, no new potassium can leave the cell, and it stops hyperpolarizing. No, like I said, that works. Stick that in there. Perfect. All right. So voltage-gated sodium channels close right away as soon as we pass the threshold. Voltage-gated potassium, just like they open slowly, they close slowly as well. However, once closed, or let's be more specific, once the voltage-gated potassium channels are closed, the neuron can use pumps and leak channels to repolarize back to the resting membrane potential. Right, and I guess we stopped counting. So 4 was up there. This would be 5. That would be 6. I guess this makes us 7. Again, the numbers don't really matter. You know what? I'm not even going to bother numbering because that'll just be more confusing than anything else. But actually, I will just for consistency's sake. Back to the resting membrane potential. So that makes that 7. Let's cheat and go back and make this 6. That makes this 5. Oops. There. So once our, we gotta go back to red. Once our voltage-gated potassium channels close, then using pumps, oops, don't want that to be so small. Once voltage-gated potassium channels, using pumps, using leak channels, the cell will come back to and be at its resting membrane potential again. And that is our equilibrium potential. Notice we talked about neither sodium nor potassium actually get to reach their equilibrium potential. But letting them into or out of the cell allows us to produce this big, huge current, this big, huge electrical signal that allows us to communicate and spread that communication with all the different parts of our body that our neurons communicate with. And that, in its simple 7 steps, is our action potential. All right. Is this a potential essay question? I, in fact, guarantee that this will be an essay question. Let me rephrase that. There are basically three main action potential questions. This is how we produce an action potential, right? That's the key here. This is how we produce an action potential. We will also talk about how to propagate an action potential, how to spread it down the axon. And if you remember, we also talked about how myelin speeds up that propagation, how we spread it. So how do you produce an action potential? How do you spread it without myelin? How do you spread it with myelin? Those are easily 3 essay questions, and there are probably more. But I guarantee you, again, this is our big, huge process. So absolutely, there's going to be a question on this. So great question. What I will say is that if I make this an essay question, if this is an essay question, one of the things that I'll remind you, and I don't know how many of you actually took advantage of it, but because I didn't look at all of the videos, not for all of the parts of them. But if you remember, there is a drawing tool, that whiteboard. What you can do, since I'm recording the lecture, if you bring up the whiteboard and write on that whiteboard, I can see what it is. So if during that essay question, you bring up your whiteboard and you draw on that whiteboard, you can draw the actual potential on that whiteboard and then explain it on the essay question. Or even type it on that as well. So that is something that I could, so yes, I could potentially have you draw an action potential by using that whiteboard during the exam. You can describe this without drawing it, but if drawing it helps you, then that would be the whiteboard. So that would be my guess. If I had this as an essay question, I wouldn't require the drawing, because if you just want to describe it, that would be fine. But if you wanted to draw it, you could use the whiteboard for that. Great question. All right. Questions on the action potential. It really is just that simple. Okay, maybe that's not just that simple, but it is. All right, so let me go ahead and save this picture because it's beautiful and I love it. Again, I'm a neuroscientist. This warms the cockles of my heart. And let's go back to the lecture and look at it with all the pretty pictures and all the pretty words. All right, so I saved that. I don't think there's anything more we have to do with that. Let's switch back to the lecture. And the cool thing about the lecture is the lecture, like I said, has some pretty pictures in them. Notice here, we see the pretty pictures of our voltage gated channels. Where's my annotation tool? There it is. And there, perfect. So here is the channel, the proteins in the plasma membrane that allow ions to flow through it. And remember, as we talked about it, it has 2 gates. So is this, hold on, there's another question. Cool. So 2 gates. This first gate is the activation gate. These activation gates are basically proteins that are very positively charged. And then remember, as we talked about, our sodium channel has a voltage, has that inactivation gate, that second gate, that globular protein and linear protein, that ball and chain. Remember, voltage gated channels work when the inside of the cell gets more positive. Then what ends up happening is that these gates swing open. This gate swings up, this gate swings up, and now it's open and ions can flow through it and that channel is open. However, remember also with our voltage gated sodium channels, this ball has the potential to wedge itself into this channel. And when it wedges itself into this channel, it blocks the channel so that even though the gate is open, ions can't come through, and we say it is inactive. And what's really cool about this is a tremendous amount of research has gone into it. We know all molecules move around randomly inside our body. They have their own kinetic energy. And it turns out that's all that causes this ball to wedge itself in here. It's just random kinetic energy. They know that because what they've actually been able to do is go in and manipulate the length of the chain. right? If you make the chain shorter, there's less freedom of movement, and so it's more likely to wedge itself in there as it randomly moves. Where if the chain is really long, it can wobble around for a much longer period of time before it just happens to lob itself in there. So it really is kind of cool the way this thing works. But so the key is our sodium channel has 3 states. Open, closed, and inactive. Our potassium channel has only 2 states. Open and closed. And remember, the other big difference between these two is that our sodium channel opens and closes quickly, whereas our potassium channel opens and closes slowly. All right. So notice here we have our resting state. We're at negative 70 millivolts. When we're at negative 70 millivolts, both are, let's go ahead and erase all these drawings that I've done so there's no confusion. Both our voltage gated sodium and our voltage gated potassium channels are closed. Then, using a graded potential, that graded potential spreading down the dendrites, spreading down the cell body to the axon hillock, depolarizes it. And if it depolarizes it to threshold, that's when the magic is going to happen, right? Remember, as we talked about, an action potential is an all or nothing event. You either get an action potential or you don't, right? You're either pregnant or you're not. It is all or nothing. And so if you reach threshold, you get an action potential. You don't reach threshold, no action potential. And remember the other key, threshold is negative 60 millivolts for all of the channels, both the sodium and the potassium. That light switch pressure is the same for both of them. The difference is how quickly they open. Right? Erase that. So, step 1, reach threshold. Step 2, depolarization. Voltage gated sodium channels open. And remember, we have a massive amount of these. So a massive amount of voltage gated sodium channels open, and a massive amount of sodium enters the cell. And a positive ion entering the cell makes the cell more positive. We get a big, rapid depolarization because sodium's coming in to try to get the cell's membrane potential to equal positive 66 millivolts. So we have a massive amount of sodium coming in. All right? It wants to reach its equilibrium potential, but remember, it's not going to get there. It's going to get close, but it's not going to get there. And the reason it doesn't get there is 2 things happen. The first thing that happens, as we see here, is that pesky ball has wedged itself into the channel. Our channel is now inactive. And if it's inactive, remember the key to this is it cannot open again until it closes first. Right, as we saw, it must close to kick the ball out. Right? So it has to close to kick the ball out. Once it's inactive, it's going to stay that way until the channel closes. That means no new sodium can come in the cell, and the cell can't keep getting positive. But the other thing that happens is finally those slow voltage gated potassium channels have opened. Once they open, that allows potassium to leave the cell. So the cell was really, really positive, rapid depolarization, but it reached a peak because sodium stopped coming through, so it couldn't get more positive. And as all this positive potassium leaves, the cell starts to get more negative. And again, it is going to repolarize and hyperpolarize. Right, potassium leaving causes the cell to repolarize and hyperpolarize. Because again, it's trying to reach its equilibrium potential of negative 90 millivolts. Negative 70 is just on the way. It doesn't care about resting membrane potential. It just happens to be going that way. All right, so this starts that repolarization and ultimately that hyperpolarization. So again, potassium rapidly exits the cell repolarizes. But remember, and let's cheat and draw a line on here, right? On the way down, it passes threshold. And at that threshold, and notice this beautiful picture does such a great job of showing this, at threshold, that channel closes. When it channel closes, it kicks that ball out. The channel is no longer our voltage gated sodium channel. It is no longer inactive. But remember, potassium channels close much more slowly, so they stay open for a longer period of time. And so the cell keeps moving towards the equilibrium potential of potassium. The cell hyperpolarizes, and as we mentioned, we call that the undershoot. But eventually, with a delay, those potassium channels are going to close. And once they close, the cell uses pumps, the cell uses leak channels, and the cell goes back to its resting membrane potential. And when you put it all together, it looks like this. Notice your book described it in 4 steps. I think we used, I don't even remember now, was it 6 or 7 or something like that? I don't remember exactly how many we moved. As I have often said many, many times, I don't care how many steps you use, as long as you describe all the information in all of the correct order in all of the appropriate time. That's what I care about. All right. I want to show you a couple more pictures, starting first with this one. I really like this one because this one, and I'll be honest, you've been using this for so long, I don't even remember if this one comes from your textbook or not. But what I really like about this one, and we'll cheat and draw over the top of all of these, we'll go ahead and use the highlighter so that we don't block anything. It shows first the action potential. So notice again, we are coming up, we reach threshold, we get that rapid depolarization, that rapid repolarization, that undershoot, and then it comes back to rest. So that's the action potential we've talked about. But what's cool here in yellow and green is here in yellow and green, these represent the permeability of the cell to the ion, right? Basically, that's a fancy way of saying how much of the ion can move across the plasma membrane. Notice at first, and let's go ahead and we'll start with our green, our yellow for orange for sodium. Notice at first, right, all those voltage gated sodium channels are closed. But notice, as soon as the cell reaches threshold, all of those sodium channels rapidly open up. And when all of them open up, massive amounts of sodium are coming into the cell. But then, just as quickly, that ball of the ball and chain wedges itself in there. And when the ball of the ball and chain wedges itself in there, suddenly sodium can't enter anymore. And so just as quickly, the permeability of sodium goes back down to 0. Notice on the other hand, potassium, again, it starts at 0, but when we reach threshold, they open. But notice they open much more slowly. It takes them a much longer time for them to fully open. In fact, if you notice, by the time they've all fully opened, you've practically reached the threshold on the way back down, and they are going to start closing again. Right? So it's really just before they reach the threshold on the way back down, and then they start closing again. But even the same way they open really slowly, they close really slowly, so their permeability stays up for a longer period of time, and that is what causes that undershoot. But eventually it goes away, and then the cell is able to repolarize. So this is kind of a really fun illustration that kind of shows the differences in the speeds of the opening and the closing of those channels and how they affect the permeability. Because sodium, a lot of sodium comes in super fast, but then it stops. Sodium takes a lot longer to be able to come out, and then it stays open for longer as well. All right. I have one more pretty picture to show you, but I'm going to cheat before we do that and actually go back, because I want this picture first. All righty. Now, as I mentioned, our goal was to produce an action potential. And that is what we have done so far. That is what we've described. And as was mentioned, that is definitely one of the potential essay questions. But as we learned with our muscle system, that typically when a neuron communicates to a cell, it sends many action potentials. So that means our job is not to just produce one action potential, but to produce many. Or at least more than one. The problem is there is one more characteristic that our neuron shares with our muscle cell. Neurons have a refractory period. In fact, neurons have 2 refractory periods. Remember, refractory period, when we were talking about a muscle cell, we said for a brief period of time, oops, don't want this to be capital. The cell loses the ability to produce an action potential. All right? And neurons have this as well. Now, here's where we need to do a little bit of critical thinking. Let's see if we can figure this out. What actually produces the action potential? Can anyone hazard a guess is, of all the events that we talked about, what is truly the one event that actually produces the action potential? Not the refractory period. The refractory period, you're absolutely correct, is going to be due to that inactivation gate. But what actually produces the action potential? What's actually, remember, an action potential is a big positive signal. So what is the actual event that occurs that causes a big positive signal? Think back to the drawing. Well, true, the graded potential triggers the event. You're absolutely correct. We're getting closer. The graded potential gets us to threshold. And what happens at threshold? that gives us the action potential. Yep, the action potential is the depolarization. But what's the actual event that gives us that depolarization? You guys, you've got it surrounded. Yep, ions crossing. And specifically what ion? There you go. Bingo. Sodium. What actually produces the action potential is when our voltage-gated sodium channels open. When that voltage-gated sodium channel opens, all those other things you guys talked about happens. We get a massive amount of the sodium ion entering into the cell. We get that big depolarization. So what this means is if we want an action potential, we have to open the voltage-gated sodium channels. Make sense? So let's think about this, and let's go over this with our highlighter. Yep, kind of like in muscles, absolutely. So right here at threshold, we cross threshold. And when we cross threshold, this event right here, this point right here, is when our voltage-gated sodium channels open. All right? Now, if our voltage-gated sodium channels are open, can you open them again? Right? If you're like me in your bedroom or in your living room, or heck, go to your back door. Go to your back door and open your back door. Once you open your back door and it's open, can you open it a second time? No. Right? So if you think about it, while the door is open, we can't open it a second time. And if we can't open it a second time, we can't get an action potential. So if you think about it, for all of this part right here, our voltage-gated sodium channels are open, are already open. And if they're already opened, we can't open them a second time. And then what happens to our voltage-gated sodium channels up here at the top? They don't close. What actually happens to them? They don't close. They're not open. There's that third state. What is that third state again? Inactive. There you go. Our voltage-gated sodium channels are inactive. And remember, as we talked about, if they're inactive, they cannot open again. Because that pesky ball of the ball and chain has wedged itself in there. So notice, for all of this period here, so basically from this period of time right here to this period of time right here, either our voltage-gated sodium channel was already opened or it was inactive and cannot open again. So notice, for this period of time, it cannot produce, exactly, so we only get one action potential at this period of time. So for this period of time, no matter what we do, we can do anything we want. But no matter what we do, we cannot produce a second action potential during this time. That makes sense? We comfortable with that idea that during this time, no matter what we do, this period of time here in green, no matter what we do, we can't produce another action potential. And so we actually call this period of time the absolute refractory period. Because there is absolutely, positively nothing we can do to get this cell to fire another action potential. It's impossible. Nothing can be done. Once the door is open, we can't open it a second time. Once it's inactive, we can't open it a second time. So for this period of time, we have this absolute refractory period. This absolute refractory period is this period of time where absolutely positively, no matter what we do, we cannot get it to fire another action potential. With me so far? All right, so, but remember I said there's 2 refractory periods. Because again, notice, and let's change colors here to, we'll use purple. At this point right here, right, we cross threshold. Oh, that's way too big. First, let's turn off that, and let's change the size. So right here, we cross threshold on the way down. And remember, at this point right here, our voltage-gated sodium channels close. So at this point right now, once they close, once you close the door, can the door be opened again? I'll just assume those yeses you guys already printed were the same rest as you're answering here. Absolutely. Now that the doors are closed, you can open them again. All right? So absolutely, you can again. And to produce, not with another action potential, if we open them again, we will produce a second action potential, right? This is about producing a second action potential. So if we can get them to open again, we will produce a second action potential. Hold on, what is this? So the ball and chain is indicating the inactive state, making is, so yes, so when the channel is open, when the channel is inactive, yes, during that state, it can't be opened again. So that what that means is we cannot produce a second action potential. And so that is our absolute refractory period. So that is the first refractory period. The first refractory period is this absolute refractory period, where no matter what we do, we cannot get another action potential. Notice, once the channel closes, we can produce a second action potential. But if you notice, there's a problem. And I'm going to go back to highlighting in purple to show this. Notice that, yes, we've crossed threshold on the way down, but notice the cell is getting more negative. The cell is getting more negative. It's getting down to resting membrane potential. It's getting below the resting membrane potential. Remember, to reach let's say it this way, to produce a second action potential, we have to reach threshold. We have to get up to threshold to be able to produce a second action potential. If the cell is down here, and let's use simple numbers, the cell is down here at negative 80 millivolts. Heck, let's go even easier than that. Negative 75 millivolts. Remember, at the first one, and feel free to ignore, here, let's in fact even cheat. I want to, boop. All right, there you go. Threshold is negative 60 millivolts. So remember here at the very beginning, we needed a plus 10 millivolt change to reach threshold. If the cell is down here at negative 75 millivolts, can we give it that same plus 10 millivolt signal and get it to reach threshold? Well, changing it 10 millivolts at negative 75, get it to threshold? No. So notice Once we close the channel, technically it can be opened again. But the key here is that it can be opened again, but we need a stronger signal to reach threshold. So notice starting at this point right here, when it closed, all the way to when the cell gets back to resting membrane potential. Notice during this period of time, when the cell is negative, we can reach threshold, but we can get it to open again. We can't get threshold, but it takes a bigger signal. And this period of time we call the relative refractory period. Notice again, these refractory periods have nothing to do with the first action potential. It's kind of like twitches. Remember when we looked at twitches? A twitch has a refractory period, and that refractory period had nothing to do with the twitch itself. It was all about when can we produce a second twitch. And it's the same thing here. Notice we didn't talk anything about refractory periods when we talked about the action potential. Instead, the action potential is an own thing. Once it starts, you get an action potential no matter what. What these refractory periods refer to is what it takes to produce a second action potential. During the absolute refractory period, there is absolutely nothing you can do to produce a second action potential. During the relative refractory period, you can reach threshold, but it takes a stronger signal. So it is just relatively harder. You can get an action potential, it takes a bigger signal. It's harder to do. Whereas in the absolute refractory period, there is absolutely nothing you can do. Now, I have made an absolute mess of this picture, but if you'll notice, and I'll go ahead and clear this. Well, actually, that might be worthwhile to keep. Let me go ahead and take a screenshot of that. Now I can clear it. If we move forward, you can see, here's the pretty picture from your textbook that shows the same thing. Notice once you stimulate it, once you pass threshold, all the way to hitting threshold on the way back, there is nothing you can do to get a second action potential. While the first action potential is going, you cannot get a second action potential, no matter what. No, there's just one absolute refractory and one relative refractory for this action potential. Now, when you produce, if theoretically you produced a second action potential here at this point, so if you produced an action potential here, then yes, it's going to have an absolute refractory period and it will have a relative refractory period as well. So yes, every action potential you produce will have both an absolute and a refractory period. So yes, you are correct. That is the case. Yes. Every action potential you produce will have an absolute, where no matter what you do, you cannot get another one, to a relative, where you can produce another one, but it is harder to do. And that's really the key. Absolute refractory period, no second AP, no matter what. Relative refractory period, yes, second action potential. But you need a bigger stimulus. And that's really the keys to the definitions of those refractory periods. Awesome, and awesome questions. Well, you need a bigger signal. So if you think about it, again, if the goal, and let's draw a line here. I gotta move this out of my way. If this is threshold, right? If you think about it, the size stimulus you need from rest to get to threshold versus the size stimulus you need from it being hyperpolarized to get to threshold, this is a much bigger signal. So notice it takes a bigger stimulus, a bigger signal. Instead of just being a 10 millivolt change, maybe it's a 20 or 30 millivolt change. So it takes a bigger signal to reach threshold. So it takes a bigger stimulus to reach threshold. And because remember, to fire an action potential, we have to reach threshold. All right. Excellent. That, just that simply then, is our action potential and the refractory periods of the action potential. And describing and explaining the refractory periods is probably another good potential essay question. So that's at least 4 we'll have right there. Your book, and again, I guarantee one of those will be on, one of those 4, at least, if not more, will be on your exam. There definitely will be some action potential questions, at least 1, if not more. Because again, that's our big functional physiological process of this. All right, how are we on time? We are actually doing good. Excellent. All right, so here's the nice table from your textbook talking about the gradient potentials, talking about the action potentials. But remember, so far we have still only just produced the action potential right here at the axon hillock. What we have to be able to talk about next is how we are going to spread that action potential down the axon. And that process, which we will explain next, is going to be propagation of the action potential. However, now is a good break point. So any questions on producing an action potential before we take our next break? All right, then in this case, we will go ahead and take our break here. We'll take a 10-minute break. That means coming back at 2:43. And at that point, we will restart the recording. So I'll go ahead and pause the recording now. All righty. So the last little bit of information we need to cover today is now to, from a lecture standpoint, is to talk about the propagation of our action potential. Again, we've been, and let's go ahead and draw it. Here's our cell body. Here's our dendrites. And here's our axon. And like I said, we know at the end that axon is going to branch and form the axon terminals and have the synaptic end bulbs at the end and all the other fun things we have talked about. And as I mentioned, up to this period of time, we have been measuring what's been happening at our axon with that voltmeter just at the axon hillock. But, and of course here at our axon hillock is where we are going to produce our action potential. We don't have to say R. Produce the action potential. But the second step of this process, the second thing we need for communication is we need to then now be able to propagate. So number 1, we needed to produce our action potential. Now number 2, we need to propagate. And again, I keep using the word propagate because, well, quite frankly, it's the correct word, but it's just a fancy word of saying spread down the axon. All right. Now, we could, of course, do this passively, like a wave in a pool. And one of the important things to remember is that electricity moves a lot like water. so we could produce a wave, a big wave at the end, and have it spread. The problem with things spreading passively is when they spread passively, they diffuse, they attenuate, they get smaller. And we don't want that to happen. We want to be able to produce an action potential and have it go however far we need it to go. If we had an axon that went from here to Seattle, Washington, we could send a signal to grandma who lives in Seattle on that action potential. And we want to be able to get that whole way. So one of the keys to our action potential propagation is not only do we need to spread it, but along the way, the action potential is going to self-strengthen. It is actually going to continue to stay big the whole way. The key to this is the action potential does not attenuate on the way. It stays the same size the whole way. All right? That would be really novel if we could do that. If you could produce a wave in a pool and it reached the other end of the pool and it was the exact same size that it was when you produced it, right? And that's not what happens. So this is something that is very, very special. So we'll do it with the pretty picture from your textbook. But once again, I think it is easiest if I actually draw it first and we talk about it first. So what we're going to do is we're going to take, basically, here was my square. We're going to take the first part of our action potential here, and we are going to enlarge that and bring it down. And that's what we'll show here. So what we need here that is our axon, and we'll even put a little bit of the cell body here. So of course, this over here is our cell body. And this right here is our axon hillock. And then of course, we have, and actually let's put the labels down here. And then we have the rest of the axon. Okay. Now, as we've talked about, starting at the trigger zone and continuing the entire length of the axon, what makes the axon so special is that it has those voltage-gated sodium and voltage-gated potassium channels that travel the entire length of the axon. And I know these are not uniform and they're not equidistant, but because I'm just trying to do this quickly, but you get the idea. These are our channels. And we know that there is a transition of these in our axon hillock. All right, now the important thing to remember is that on our axon, it has a ton of voltage-gated sodium and voltage-gated potassium channels. All right, that's how we produce the action potentials. However, what we need to remember is that to actually produce the action potential, really all we use is the voltage-gated sodium channels. So when we talk about propagation, really all we focus on, and we'll consider these that I've drawn, is just the voltage-gated sodium channels. All right. We know they have to have the potassium channels. That's how we get the whole action potential and all that kind of stuff, the repolarization, the hyperpolarization, the repolarization, all the things that go along with that. But when it comes to propagation, the production of the big wave is that voltage-gated sodium channels, and that's really what we are going to focus on. All right. Does this anatomy make some semblance of sense? No one said anything since I started the recording and came back, so I just want to make sure you're still there and alive and listening. Excellent. I got one yes, so at least one person is there. Perfect. And there's some more. Great. All right. Excellent. So let's start where we began the last process. So let's switch colors now. We will switch to, we'll start at the beginning with red and change my line to red as, oh, no, I didn't want to change that to red. Oh, well. I guess it doesn't matter. I don't care anymore. All right, change my line to red, change my text to red. Perfect. This is where we're at. So remember, this starts with our graded potential. That graded potential spreads to the axon hillock. And here at the axon hillock, we depolarize the axon hillock to negative 60 millivolts. All right. Of course, when that occurs, our voltage gated sodium channels open, and we have a massive influx of sodium, and we get our big positive signal. All right, we all comfortable with that? Nothing really new there. We opened up voltage gated sodium channels. Those voltage sodium channels opened. A massive influx of sodium occurred right here, where we have a ton of sodium. And here's another sodium. And here's another sodium. And so on and so forth. All these sodium are rushing inside. They're, of course, all positively charged. And we get this big positive signal. All right? And that gives us our action potential, right? And as we talked about, that action potential is a, I need to move that a little bit out of my way. So again, we brought this down here, do that way. That current produces a 100 millivolt, positive 100. Oops. Ugh. Change. Excellent. There is absolutely positively no new information on the board right now. So hopefully all of this makes sense, because this is all what we just finished spending the last hour talking about. How we produce that big 100 millivolt positive signal by sodium rushing into the cell here at our axon hillock. All right? Everybody comfortable with that? Any confusion on this? Any concerns about this? Questions on this? Again, this is what we just covered, so hopefully this makes sense. But now we have to talk about what happens from here, and that's where this gets a little different. So any questions before we move forward? Everybody got this? Excellent. That's what I like to hear. Now, again, it's a big wave, and these things move like water, right? So when you think about it, I take that rock and I dump it into the side of the pool, or that car into the side of the pool, and I get a big, huge wave right there where I produced it. Here we got the same thing. We have a massive amount of sodium that comes in, and that sodium that comes in produces that big wave. But just like in the pool, the big wave doesn't stay here. And so while all this sodium comes in, what's going to happen is that our sodium is going to spread. Some of the sodium is going to spread back to the cell body and to the dendrites, and quite frankly, doesn't do anything there. But some of the sodium spreads to the next segment of the axon. And when it spreads to the next segment of the axon, right here in this segment of the axon, this next segment receives a little bit of sodium. And of course, sodium is a positive ion. So when that positive ion spreads to the next segment, this next segment depolarizes. And it doesn't take a lot. A little bit of sodium spreads to it, and this next segment is going to depolarize to negative 60 millivolts. And if this next segment right here spreads to negative 70 millivolts, what is going to happen to these voltage gated channels right here in this next segment of our axon? Nope, we're not to the axon terminal yet. We're just right here. We're now right here and the very next part of our axon. So this part right here was at negative 70 millivolts. Now it's at negative 60 millivolts. What's going to happen to these voltage gated sodium channels that are right here in this part of our axon? They're not inactive yet because they were at rest. They were at resting membrane potential. These back here are red ones. These red ones, once they open, they're going to inactivate. But exactly, these were at rest. So what happens here is these now new, because that's what these are, these new resting, oops, want to change colors. These brand new resting voltage gated sodium channels are going to open. And when these channels open, not surprisingly, we get a massive influx of sodium. So what ends up happening is we get this huge, no, not red. We get this massive influx of a whole bunch of new sodiums. Na, and another Na, and another Na, and another Na. We get this massive influx of sodium. And so right here at this location, we get a 100 millivolt big positive change. Notice what we've done in reality is produced a second current. And really that's the key. Notice what really happened is here we produced a small current, and that current is going to produce a second current that also is going to spread. So what actually has happened is we've produced two separate currents. However, if you were looking at this and measuring this for the eyeball, what you would see is what looked like you had one action potential that started here, and that action potential spread to the very next spot and was the exact same size. Did not get any smaller. Of course, at this point here, the exact same thing is going to happen again. Some of the sodium spreads. Now notice, sodium will again spread in both directions. Some of the sodium, and because I'm running out of space, I will write it over here to the side. Again, some in black. Some sodium spreads backwards. So when it spreads back to this location right here, And I'll highlight this in my yellow highlighter now. When this sodium spreads back to this area right here, do I get another action potential at this point and they just bounce back and forth between these two locations? No, somebody already said the key word. This spread that spreads back here has no effect because the axon hillock is inactive. That inactive period stops the action potential from going backwards, right? And that's really one of the keys to the action potential, inactivity. That inactivity stops it from spreading. Well, it doesn't stop it from spreading backwards. It still spreads backwards. But when it spreads backwards, it doesn't produce a second action potential. Instead, the important part is we get that new sodium spreading to the new area. And of course, when it spreads to the new area, this new area reaches threshold. And when this new area reaches threshold of negative 60 millivolts, here, new voltage gated sodium channels open and sodium rushes in. And we get a big positive signal and our action potential has moved again. Right? So again, now at just this location, these voltage gated sodium channels are going to open. We have a massive influx of sodium, all of this sodium entering. I'm just going to write sodium one time this time. All this sodium coming in. And again, we are going to get a big, whoops, there, and then there, that big plus 100 millivolt change. And it looks like our action potential has continued to spread down. And this process continues. I can keep changing colors. I can keep doing this again and again, but you guys get the idea. This is going to produce a local current, and this local current is then going to open new channels, and that new channels are going to allow new sodium to come in. And when that new sodium comes in, it spreads. And when that new sodium spreads, it produces an action potential and reaches threshold. And when it produces that threshold, that action potential, that threshold, those new voltage gated sodium channels open and new sodium comes in and that new sodium spreads and again and again and again. And ultimately what ends up happening is we get a spread of an action potential down the axon that always stays at a positive 100 millivolts. It never gets smaller, right? And that's the key. That's why we can send this signal all the way from, I guess it helps I spell always correctly. We can, if we had an axon that went all the way from here to Seattle, Washington, then when we get to Seattle, Washington, we can send that action potential and it would make it that entire way and it would never change in size. And so we could send it all the way from here to Seattle, Washington. No, remember at the neuromuscular junction, our depolarization causes the release of a chemical signal. So remember, our neurons produce and send an electrical signal down the neuron. But remember, at the end of the neuron releases a neurotransmitter to talk to its target cell. But what that does tell us is that action potential that reaches the synaptic end bulb is going to be a full action potential that reaches, so where's my drawing? So down here, we're going to get that full 100 millivolt action potential here at the synaptic end bulb, which is definitely going to get us to threshold to allow that calcium to come in, and that calcium causes the release of that neurotransmitter. Right? we actually know what happens at the end because we talked about this with our muscle system. And notice, it goes one way thanks to that inactivity. The one thing I will say, and it's kind of cool if you think about it, if you were, and I don't think you actually do this on your PhysioX, you do manipulate some neurons in other ways. But if you think about it, if you had a neuron like this one up here, and you artificially with an electrode stimulated it right here in the center, because it was all at rest, what would actually happen is you would actually produce two action potentials. One action potential would spread this way, and one action potential would spread that way, right? Because since you're artificially stimulating it here in the center, both sides are at rest, neither sides are inactive. So when it spreads, it will reach threshold, and you would send an action potential in both directions. Here, it doesn't go back, right here under the normal function, the action potential does not travel backwards because of the refractory period. And so because of that, our action potential just travels in one direction. So you guys, I love where you guys are going with this. Absolutely, you have the right idea. The next thing that we have to do is come down here to the end and talk about how it communicates with its target cell. The good news is how it communicates with whatever that target cell is the same regardless of what that target cell is. If that target cell is a skeletal muscle cell, which we've already talked about, or whether it's a gland, either like a salivary gland or even endocrine gland, or even another neuron, how it's going to communicate with it is going to be the same. And we kind of already learned that. So yeah, so we will talk about that next. But right now we have to spread it down the axon first. No, the artificial stimulating of a nerve in the middle would not be something that normally happens. That's not normally how we stimulate the nerve. It's something you can do in a laboratory. And so artificially, you could get it to go both directions. So we see, that's how we see and understand how it works. But that's not how our neurons work. Remember, our neurons receive information. That's not the shape I want. Neurons receive information at the cell body and the dendrites, and then they spread it in one direction down the axon. No, and again, if we're talking about propagation, propagation is not what happens at the axon terminal. What, in fact, I'll tell you right now, what happens down here, and this, you're right, this is a potential essay question as well. How it communicates with its target cell, we actually call that synaptic transmission. So synaptic transmission is a separate question, separate possible essay question. So all we're talking about when we're talking about propagation, right, this propagation question is just how does the action potential, and that's really the key, how does the action potential get down the axon? And along with that is how does it stay the same size? All right. So that is our propagation. And again, notice this is on an unmyelinated axon. And that's important as well, because as we talked about, most axons are myelinated. And the reason for this is a very consistent process. Once you start it, it's going to go, and like I said, it could travel all the way from here to Seattle, Washington. It can go that far, which is awesome. But that doesn't mean that's what you want to use to have a conversation with grandma. Because while it is a very consistent process, if you think about it, it takes a lot of time. right? We have to have uh, that graded potential that spreads to open voltage gated sodium channels, have the sodium rush in, some of it spreads down to open more channels to allow sodium to come in, to spread down, to reach threshold, to open more sodium channels, to allow more sodium to come in, and again and again and again. And notice, none of the first sodium ions that came in are going to make it down to the end. It's always new sodium that's coming down. And it's always the same size, and it's very consistent. But the key problem with this is that it is a relatively slow process. Absolutely positively, I could send a action potential along an axon from here all the way to Seattle, Washington, if I wanted to communicate with grandma. But here's the problem. My action potential would take 2 days to get there. So if I said hi to grandma by action potential, it would be 2 days by the time she got that signal. And then anything she tried to send back to me would take 2 days to get back to me. All right? Grandma's likely dead before we finish this conversation, if that's the way this is going to work. So it is a relatively slow process, but we need to be able to make it faster. And the way we make it faster is with our myelin sheath. All right. So if we understand how to propagate on an unmyelinated axon, let me go ahead and take a picture of this. And then I'm going to erase this and we'll look at the pretty picture from your textbook. And then we'll see if we can't speed things up with our myelin. So clear that. Bring back my chat window, my annotation tools. Excellent. So notice here, a little bit prettier, is the same type of picture that we were just looking at before. Here's our axon hillock. Here's the first part of our axon where that trigger zone is located. Notice we got it to threshold with a graded potential. We opened voltage-gated sodium channels, and voltage-gated sodium rushed into the cell, bringing the cell to positive 30 millivolts. Once that occurs, some of that sodium is going to spread, producing that local current, and it spreads to the next segment. When it spreads to the next segment, notice all of it doesn't have to spread. We don't need all 100 millivolts spreading. We just need a tiny bit. We just need enough to get it to threshold. And if we can get that next segment to threshold, its voltage-gated sodium channels open, new sodium rushes in these new channels, and in the next segment, we get a big 100 millivolt change. We get our action potential. Of course, this then spreads as a local current to the next one, reaches threshold, opens those channels, and the process continues. Incredibly consistent process. Very, very consistent process. But, like I said, it is relatively slow. To send a signal from here to Seattle, Washington would take 2 days. Now, if this takes 2 days to get from here to Seattle, Washington, how can we possibly use this in our own body? There's no way this could be possible. Now, we did say most of the axons are myelinated, but most is not all. So how can we possibly have any axons that are unmyelinated? And how can they be successful at helping us in our body if it takes 2 days to get from here to Seattle, Washington? Anyone want to hazard a guess? Well, how long are the axons in your body? Are any of the axons in your body long enough to get from here to Seattle, Washington? No, of course not. Absolutely. Remember, we said your longest ones are like 3 feet, a meter in length. So yes, it is a relatively slow process, but our axons are relatively short. So yes, there is a bit of delay, but that delay is fractions of a second. You can actually measure this. We actually have some reaction time rulers in the classroom we can use, but you can do a web search. There's all these different types of reaction type things where you look at a stoplight, you see a green light, and then when it switches to red, you have to click the space bar or something like that, and it measures your reaction time. Many of us have done those types of things before. A lot of that delay in the reaction time is the time that it takes for that signal to spread along the axons, right? We have to receive, for the visual information has to get to our brain, then we have to process it, so there is a little processing time, and then we have to send a signal out to our hands. So some of it's processing time, but some of it is also the delay of that signal getting out to our fingers. But since our axons are relatively slow, the delay is relatively short. We're talking fractions of a second. However, that doesn't mean it can't get better. And to get it better, we use myelin. Myelin sheath produces something we call saltatory conduction. Saltatory conduction is just a fancy way of saying jumping, because that's basically what it looks like our action potential is doing. It looks like our action potentials jump from one spot to the next. Now, of course, saltatory conduction requires myelination. And of course, as I'm sure you remember from the last class, the cells that are responsible for that myelination in our central nervous system are the oligodendrocytes. And in the peripheral nervous system are the Schwann cells. And notice, as you see here in the illustration, remember the myelin doesn't cover the whole axon. There are gaps in between, and we called those gaps the nodes. And again, they are the nodes of Ranvier, which, like I said, is tremendously fun to say, but you don't actually have to say of Ranvier, you can just say the nodes. All right, I am going to cheat now that we looked at the picture here. Oh, yes. No, great question. Gaps are in both the peripheral nervous system and the central nervous system. The only difference between the myelin on our central nervous system and our peripheral nervous system are the cells that make it. But both need gaps, and we're going to see why those gaps are so important right now. Now, to do that, I'm going to cheat and put a big white block over the slide so that I can draw on top of it. And I'm going to draw what we looked at before. So here is our axon, just like before. And again here, and I'll just abbreviate it, is our axon hillock. But what we then have, as we've talked about, are these big chunks of myelin sheath. And now that I've done that, I think I need to shorten it just a little bit so that I can put a second one right here. About the same length, good enough. And then, of course, remember, it wraps all the way around. So let's put myelin on this side as well. All righty. Now, as we know, at the axon hillock, we have those voltage-gated sodium and voltage-gated potassium channels. So again, I'm going to write here, we have voltage-gated sodium and voltage-gated potassium channels. But remember, I'm going to put the voltage-gated potassium in parentheses, because when it comes to propagating our action potential, all we really care about are the sodium. Okay, so I'll remind us of that right there. Now, notice, would it do us any good to put a voltage-gated sodium or a voltage-gated potassium here along the axon where the myelin sheath is located? No, because remember, this myelin sheath is comprised of hundreds of layers of the plasma membrane of that cell. And if you think about it, plasma membrane is a phospholipid bilayer. So basically, there's a massive amount of fat, hundreds and hundreds of layers of fat that are wrapped around the axon in this point. So there is absolutely no reason. Oh no. Oh, I erased that. Okay, let me get that to go back. Perfect. I'll cheat and do it this way. There is no reason to put those voltage-gated sodium or voltage-gated potassium channels here under the myelin sheath. There'd be no place for it to go. But if we want to continue to produce action potentials, we need them. And so luckily we have our voltage-gated sodium and voltage-gated potassium channels here in our node. So right down here, I'll remind us that this is one of the nodes, and over here is a second node. And in these nodes is where we have our voltage-gated sodium and voltage-gated potassium. But again, all we care about right now are the voltage-gated sodium channels. All righty. Are we comfortable with the anatomy of this? Any questions on the anatomy of this before we move forward? All right, excellent. So this is going to start the same. All right, oh, and let's change the color of this. I think we started with red last time, so let's stick with red. We have a graded potential that comes to the axon hillock. We reach threshold. Our voltage-gated sodium channels open. And we get a massive influx of sodium. Right? So again, here, these channels open. We have a huge influx of sodium. And we get, as a result of that, a big positive 100 millivolt change, right? Our action potential. Great question. So again, there is no difference in the myelin sheath of the central nervous system and the peripheral nervous system. The myelin sheath functions the exact same way. In both the peripheral nervous system and central nervous system, they wrap around the axon hundreds of times, producing this special insulation that is going to allow us to speed up our action potential. So functionally, they do the exact same thing. Literally, the only difference between the myelin sheaths in the central nervous system and the peripheral nervous system are which cells wrap around the axons hundreds of times. In the central nervous system, it is an oligodendrocyte. In the peripheral nervous system, it is a Schwann cell, right? Think of it as a union thing, right? If you are a delivery person, your job is to deliver something. If you drive your truck as a delivery person to a dock where you are going to then deliver that food, you can't unload your boxes at that dock, right? Instead, there are delivery people there, Teamsters, whose job it is to deliver things. You're both delivery people, but Teamsters are a very strong union. They control the docks. Truck drivers have their own union that control the trucks. So you're both doing the exact same job. Both of your jobs to do something. The only difference is you're in different locations. Right? These things do exact same things, but in the central nervous system, it is the oligodendrocytes that wrap themselves around, and in the peripheral nervous system, it is the Schwann cells. Now, the only other difference is, remember, one oligodendrocyte can form myelin on dozens of pieces of axons. So they can myelinate multiple axons of multiple neurons, whereas one Schwann cell wraps its entire body around just one segment. So if this was a peripheral neuron, this right here would be one Schwann cell. But other than that, they're completely different. Myelin sheath works the same regardless of which cell produces it. Does that help? Excellent. All right, so we produced our initial action potential. This was exactly the same thing that happened before. And just like before, sodium is going to spread. And again, let's use really simple numbers. Since it's a 100 millivolt change, let's say that 100 sodium entered the cell. Of course, it's not that. It is way, way more than that. But again, I'm going to go with really simple numbers. And as we talked about, some of that sodium is going to spread. Some of the sodium is going to spread backwards. And some of the sodium is going to spread this way. When the sodium spreads this way, it's not going to be all of the sodium. So let's say, for instance, right here, only, let's say half, 50 of the sodium spread to this part right here. Normally, when it spreads, 50 would be more than enough to give us to reach threshold and open more channels, but there are no channels to open here. But just like when you produce a wave in a pool, it doesn't move to the next section of the pool and then just stop. It's going to continue to spread. So in this case, what happens is our sodium continues to spread under the myelin, which is fine. It's totally fine that this is spreading under the myelin. However, as we talked about, this is a passive spread, like the spread of water in your pool. Notice we're not adding any new sodium to it on the way. And so with this passive spread, as it spreads, it gets smaller. And again, remember the fancy word we gave for that is it attenuates. So what happens is our signal spreads and it gets smaller. First 50, and then maybe there's only 40 sodium, and then from there, there's only 20 sodium. Oops. And so on and so forth. And then finally here, 10 sodium reach the node. Oops, doesn't need to be capitals. So notice our signal is getting smaller along the way. We started with a 100 millivolt change, and then it becomes 90, and then it becomes 80, and then it becomes 70, and then it becomes 60. Our signal attenuates. The signal gets smaller as it travels passively. However, we produced a 100 millivolt change, but we don't really need a 100 millivolt change. Remember right here at this node where we have voltage gated channels, while the signal gets smaller, we only need it to be big enough to reach threshold. And remind me again, how big does it need to be to reach threshold? Both. The sodium is the positive signal. So the sodium coming in, less sodium spreads, and so the cell gets less positive. So it is the action potential, that positive signal being produced by the sodium, that is attenuating. And how big of a signal do we need here at our node to reach threshold? let's see, resting membrane potential is negative 70 millivolts. What's threshold again? Yeah, negative 60. So what that means is we only need it to be a positive 10 millivolt change. Notice it started as a positive 100 millivolt change. Notice it can attenuate as much as 90%. And as long as there is still a positive 10 millivolt change left, then boom, we reach threshold. And if we reach threshold, all the voltage gated sodium channels open. We get a massive influx of sodium, and we produce a 100 millivolt action potential. Here at our node, we are going to now get a big, huge, oops, I drove that too big. Here we're going to get that big, huge plus 100 millivolt change. We get our action potential. And notice, it looks like our action potential has jumped to the first node. Great question. So again, let's go back over this exact same space and talk it out. Just saying the words and see what happens. All right. We open voltage gated sodium channels. A whole lot of sodium enters the cell. A lot of that sodium starts to spread down the axon, and then there's less, and then there's less, and then there's less. And then we reach the node. At the node, we have enough sodium to reach threshold, and we produce another action potential. All right, we compare that to sodium rushes in, we get a big, huge action potential. A little of that sodium spreads to the next section of the axon. In that next section of the axon, we depolarize that cell to threshold, and the voltage-gated sodium channels at that section open. A massive amount of sodium enters into the cell at that section of the axon, and some of that sodium spreads to the next. When it spreads to that next segment, it brings that region to threshold. Now a bunch of new voltage-gated sodium channels are going to open, and a whole lot of new sodium is going to rush inside, and we have this big, massive influx of sodium. A little of that spreads to the next segment, and when that little bit spreads to the next segment, that next segment is brought to threshold, and new voltage-gated sodium channels open. We get a huge, massive influx of sodium entering into the cell. Some of that spreads to the next segment, and on and on and on. Notice we have a lot more sodium ion movement. We have to open a lot more channels. We have to produce a lot more currents to keep that consistency of it spreading. Here, we just have that passive wave moving to reach from one side to another. And that's really the key to this. Because we don't have to open as many channels, we don't have to allow as much sodium to come inside, it is a much faster process. Nothing like wave summation, right? This is nothing like wave summation. This is more like dominoes, right? If you were to put up dominoes to knock down dominoes, you could literally put a row of dominoes right next to each other where they were actually literally touching. Okay? So you put 10 dominoes up where they're literally touching, you tap one, and it's going to knock into the second, which is going to knock into the third, knock into the fourth, knock into the fifth, and they're all going to get knocked down. And is that going to go a really long distance if you have them next to each other, actually touching each other when you tip them over? No, it's going to go a very short distance. However, if you take a domino, take 4 dominoes that are touching, and pull out the 2 middle ones, and then spread them 2 apart, and spread the next ones 2 apart, and spread the next ones 2 apart. If there's gaps in between the dominoes, when the first one falls, it falls further, hits the next one, and then knocks that one over. And notice in that way, you're going to get with 10 dominoes, you can get a signal a much further distance. So that signal travels a lot faster. And that's really what we're setting here. What we're doing is we're moving the dominoes further apart, because the further apart you move the dominoes, the faster the signal travels. All right? And that's great, except for one problem. What if you spread the dominoes out too far? Now the first domino falls, and what happens? It doesn't hit the second one. And the same thing can happen here. This is all great that, yes, it goes much faster as that signal is spreading passively under the myelin. But what happens if when we get here, the signal is too small? If when we reach the node, let's say the signal instead is only, oops, a +8 millivolt change, will that be enough to get it to reach threshold? No. And so what would happen is our action potential would stop. So there is more of a risk. Remember, the other one we said is slow, but it was consistent. This one is faster, but there's a little bit more of a risk. And so that's why, not surprisingly, now that we know this, I said most of our axons are unmyelinated, but one of the that are myelinated, but one of the systems that is not myelinated is your pain system. On the surface, that doesn't sound like a lot of sense. If I touch something hot, I want that signal getting to my brain as soon as possible to let me know that I'm being injured. Instead, it's unmyelinated. What that means is it's a little slower getting to my brain, but I know with 100% certainty it's going to get there. Now, these are set up, to be precise in their function so that nothing happens. And it is the right distance where we get the right speed, but we still have a small enough signal. However, what if theoretically there was some type of disease? And that disease, for instance, destroyed the myelin sheath. If that was the case, what would happen is that we would lose more signal. With that disease, without the insulation, again, think of it. If you peel the insulation off a wire, the electricity can come out. If the myelin is damaged, we lose too much signal, and the action potential could fail, or really would fail. Luckily, there is no disease that damages the myelin of our axons and causes that kind of problem, right? Or wait, or is there something like that? There you go. Multiple sclerosis. Multiple sclerosis is an autoimmune disorder where your own immune cells destroy your myelin sheath. They attack those Schwann cells in the peripheral nervous system. Pardon me, the other way around. The oligodendrocytes of the central nervous system, and cause serious complications in your ability to produce and send action potentials. So yeah, so this one is much, much faster. We're producing longer currents, faster currents, but there is that risk that the signal could potentially fail. All right, let me take a pretty picture of this one, and then we'll look at the same pretty picture from your textbook, because again, your textbook does a nice job of showing these things. So here's that exact same setup. We have our myelin sheath, we have our nodes, and we have our axon hillock. We produce that signal. That signal spreads passively, a much longer current, right? It travels faster because it's just passively traveling, but it's smaller. But it doesn't matter if it gets smaller, as it can attenuate 90%. As still as it reaches threshold, it will open those voltage gated sodium channels, allow a new action potential to come in, which spreads to reach threshold, and this process continues. And as I mentioned, this myelination can speed up the action potential up to 6 times as fast, or 6 times faster. So there's a huge, huge benefit. And so it's not surprising that most of our most of our axons are myelinated. All righty. Questions on that? All righty. With that, we are done with everything that I wanted. Oh, actually, that's not true. I guess we one last thing, since we just talked about it, we will finish up with this then. Modifying the action potential, right? We just talked about how multiple sclerosis damages the myelin, and so it can cause that jumping, that saltatory conduction to fail. But again, the other thing that we can do to stop our action potentials is to block those voltage-gated sodium channels. And this is actually what we do with some anesthetics, and there are also some toxins that do this as well. We could take advantage, and I guess I have to bring the chat window up if I'm going to ask this question. All right, so back in ancient times, right, you would play baseball, and when you play in baseball, one of the things that would happen is the pitcher could throw the ball and hit you in the shoulder. And of course, you get first base for that. Being the bigger person, you're not going to charge the mound and try to hit him with the bat, although that is your decision to make that. But what do you do when you get to first base? What do you do when someone's punched you in the arm and your arm hurts? What do you do when your arm hurts? That's right, you do nothing because you're a man and it's not manly to cry. True. Okay, yes, you don't use it. Oh, come on. Go find a loved one in your family right now and punch them in the arm and see what happens. See what they do. Okay, don't do that. I'm kidding. But if they could get mad. There is that. But after they get mad, what do? Okay, they hug it out. How about rub it, right? If someone punches in the arm, what do you rub it? Why do you rub it? Yeah, you grab it, but not only do you grab it, but you rub it, right? Heck, punch yourself in the arm right now, right? And then what happens when you rub it? What happens when you rub it where you got punched in the arm? The pain goes away, or it's less, absolutely. And the reason for that is what actually happens is when you are rubbing your arm in that location, you are stimulating the tactile sensors in your arm, and their pathway is similar to the pain pathway. So what happens is when you rub the arm, your tactile, the senses of touch in your arm, overwhelm the pain sensors, and the pain sensors, your brain doesn't receive that pain signal anymore. Of course, as soon as you stop rubbing it, then the pain signal gets through again. So you just have to keep sitting there rubbing it. But like I said, if you're standing there at first base, you can't rub your arm because that's not manly. But yeah, we rub it and it feels better, right? And the reason for that is you're basically blocking that pain signal. And that's exactly the same thing we do with topical medications, right? Lidocaine. You've got a bad back because you've been, instead of standing in front of the classroom and lecturing for 4.5 hours all day working on your posture, now you're slumped in a chair using very bad posture to give your lecture as you're sprawled out across the floor or in your chair or whatever it is that you're doing. So maybe your back's hurting a little bit more. So what do? You take one of those lidocaine patches or lidocaine rub and you put it on that. Or maybe back in ancient times when we were allowed to go to the dentist, right? And you had to fill a cavity, they would inject Novocaine into your jaw first. These things block the voltage-gated sodium channels. And so if you block the voltage-gated sodium channel, that action potential can't get through and the pain signal never gets to your brain. One of the great examples of this, and again, it is not nearly as common in the United States as it used to be. In fact, in all of, similar with spinal block. Spinal block is the same way. They either will do chemicals to block it, right? So you block the action potential go through, you get the epidural. That's what they do with an epidural when you're going to go in for pregnancy and things along those lines. So again, you're blocking that pain signal, blocking that voltage-gated sodium channel so that the pain signal doesn't get through. Another toxin is tetrodotoxin, or what is known as TTX. It is produced by the pufferfish. Again, it is not legal in the United States. It used to be legal in Canada, but I don't believe it is anymore. Well, now it's not legal to go to get sushi anywhere. But back in ancient times, and by ancient times, I mean, like the 2000s, 1990s, there were places in Canada where you could go get a very specially produced sushi called fugo. And fugo is properly prepared pufferfish. You would have to sign a waiver before you would be able to be served the dish. The chefs that prepared it had to be retested every 30 days, because the key to this is when you prepare the puffer fish, they have a bladder that contains this toxin, tetrodotoxin, that is a very long-lasting voltage-gated sodium channel blocker. The point of it is to have just the slightest amount of it on the sushi when you eat it, so that when you pick it up, you get a tingling sensation in your fingers. When you put it in your mouth, you get a tingling sensation on your tongue and on your lips. But not so much tetrodotoxin that it gets into your blood supply and, I don't know, goes to the muscles of your lungs and stops you from being able to breathe. Right? For a time there were at least a half a dozen people a year who died from improperly prepared puffer fish. Like I said, it used to be legal with the waiver in Canada. It's never been legal in the United States, or at least not for a very, very long time. I don't think it's legal in Canada anymore, so no point in going up there. But is that the fish that you have to cut at the right angle to not die from? It is one of the ones. I don't know if there may be others as well, but yes. Tetrodotoxin, again, as we learn these things, we use it. Tetrodotoxin is an important voltage-gated sodium channel blocker, action potential blocker that is often used in research because it can last for 2 to 3 days. So again, it's not the kind of thing that is used recreationally or for other things, but it is used in research. But it's another example of a way that they can block that voltage-gated sodium channel and stop that action potential. All right, with that, that is everything we need to talk to for today. Again, we went a bit long. We are going to make up some of this by doing the histology on Friday. And then the good news is we are doing good on the lecture. We're actually a teeny bit ahead of the lecture. So what that will allow us to do on Tuesday is to be able to spend more time on the anatomy. So we are going to get to the anatomy and talk about the anatomy. But I think it's important that we be laying our groundwork. So the lecture will be less exhaustive on Tuesday, and we'll have more time to instead be exhausted by the anatomy, which we will start talking about as well. All righty, so any questions on that before we call it a day? I think 4 hours is probably enough time to be talking, so I think this is a good stopping point. Any questions on anything we covered? All right, so in that case, as always, I will go ahead and stop the recording. If you have any questions, you're welcome to continue to hang out and ask them. Otherwise, I hope to see you Friday. Voltage-gated sodium. So tetrodotoxin, like lidocaine, like novocaine, all of those things block the voltage-gated sodium channels. Remember, what their job is to stop the action potential from getting from the cell body to the synaptic terminal, to the release. When we talked about the neuromuscular junction, we talked about things like botulism toxin and curare and things that can stop the transmission at the synapse. But we'll get to that synaptic transmission in the next class. So yeah, at this point, we're just stopping the action potential from getting from one side of the axon to the other. That's all these things do. They block the voltage-gated sodium channel. And if you block that voltage-gated sodium channel, that stops the signal. All right, great question. Any others? All right, excellent. Well, then, as always, you guys be safe. Have a good weekend. I hope to see you on Friday. Please, please be there. Like I said, if no one shows up, I'm not doing it by myself. So it is important to be there. And then also it'll help you get the information to complete your histology handouts, which you will now be turning in as an extra credit assignment. Excellent. All right, guys, have a good day. Be safe, and I will see you soon.